500 million years ago, a predator with compound eyes the size of serving platters stalked the ocean floor, grasping prey with spined appendages that could rip through shells. This was Anomalocaris, the largest animal on Earth at the time, and you have never heard of it. Nearby, a creature scientists would later assemble upside down for 30 years, hunted on legs, it walked with upward. Deeper still, animals with 5 eyes, spiral tooth formations that defied physics, and body plans so alien we still debate whether they were even alive. If you enjoy diving into Earth's strangest chapters, hit that like button and subscribe. We are going back, way back, to when the ocean belonged to monsters you cannot imagine. A shadow passes over the seafloor 520 million years ago. The creature casting it measures 3 feet long in a world where most animals barely reach the length of your finger. 2 appendages extend from its head, lined with spines like curved daggers. Each eye contains 16,000 lenses, giving it vision sharper than any creature before it. Anomalocaris was the first apex predator in Earth's history. Nothing like it had ever existed. While other animals grazed on bacterial mats or filtered plankton, this hunter actively pursued prey. Those frontal appendages worked like grappling hooks, seizing trilobites and pulling them toward a circular mouth, ringed with overlapping plates. Scientists once thought these mouthparts were jellyfish fossils. The appendages were classified as shrimp tails. The body was labeled as a strange sponge. It took decades to realize these were pieces of one impossible animal. The eyes alone revolutionized sight. Each compound eye sat on a stalk, rotating independently to scan in multiple directions. The 16,000 individual lenses created a mosaic image, far beyond what simple eye spots could achieve. This was not basic light detection anymore. This was hunting vision, predator vision, the kind that tracks movement and judges distance. Anomalocaris swam using flexible lobes along its body, rippling them in waves to glide through open water. Fast for its time, certainly faster than the armored trilobites scurrying below. When it struck, those spined appendages clamped down with enough force to crack shells. Fossil trilobites still bear the scars, semicircular bite marks matching Anomalocaris' mouth perfectly. But here is what makes this creature truly alien. Its mouth could not fully close. The overlapping plates created a pineapple ring shape that contracted to crush prey, but never sealed tight. This meant Anomalocaris likely could not eat hard-shelled animals whole. Instead, it grasped them, cracked them open, and sucked out the soft tissue inside. A 3-foot predator that fed like a person eating crab legs. The Cambrian seas offered no defense against this hunter. Armor was still primitive, mostly thin shells that spines could puncture easily. Speed was limited by simple body designs. Anomalocaris owned these waters completely, the undisputed top predator for millions of years. Then, the ocean changed. Prey evolved thicker shells, better armor, faster escape mechanisms. By the end of the Cambrian period, the arms race had begun. Anomalocaris and its relatives started to decline, outcompeted by newer predators with more efficient jaws and stronger bodies. But for one brief, terrifying window in time, this creature ruled absolutely. Scientists have found Anomalocaris fossils across the world, from Canada to China to Australia. The Burgess Shale in British Columbia holds some of the most detailed specimens, preserving even the delicate eye structures. These fossils proved that complex predation, sophisticated vision, and active hunting all exploded onto the scene during the Cambrian period. Before Anomalocaris, the ocean was a garden. After it, the ocean became a battlefield. The question scientists still debate is simple but haunting. What evolutionary pressure created such a perfect hunter so suddenly? What force pushed simple organisms to develop compound eyes, grasping appendages, and predatory behavior all at once? The answer might be more disturbing than the creature itself. The fossil looked like a fever dream pressed into stone. 7 pairs of rigid spines jutted from what appeared to be a worm's back, each spine sharp as a needle and twice as long as the body was wide. Between them, smaller tentacles waved in rows. One end showed a rounded bulge scientists assumed was the head. The other end tapered to nothing. When researchers first reconstructed Hallucigenia in 1977, they placed it walking on those spines like stilts, with the tentacles pointing upward from its back, possibly for feeding or breathing. They had it completely backward. For 3 decades, every textbook, every museum display, every scientific illustration showed Hallucigenia upside down. The spines they thought were legs were actually defensive armor. The tiny tentacles were legs. What they called the head was the rear end. The creature they had assembled was an inverted nightmare that could not possibly function. The correction came in the early 2000s when better preserved fossils from China revealed the truth. Hallucigenia walked on those small tentacles, low to the seafloor, with the spines protecting its back from predators above. The actual head sat at the opposite end, equipped with a simple mouth and possibly eyes, though the fossil evidence remains unclear on that detail. But here is where Hallucigenia gets stranger. This creature measures barely half an inch long, yet it terrified paleontologists for years because nobody could figure out what evolutionary group it belonged to. The body plan seemed entirely alien, unrelated to any modern animal. Some scientists suggested it might represent an entirely extinct branch of life, a failed evolutionary experiment that left no descendants. They were wrong about that too. Recent studies using electron microscopy to examine the fossils in extreme detail revealed something shocking. Hallucigenia has claws. Tiny paired claws at the end of each leg, almost identical to the claws found on modern velvet worms, a group of land-dwelling invertebrates that still exist today. This half-inch Cambrian nightmare is their ancestor. Velvet worms live in tropical forests now, hunting insects by shooting sticky slime from glands near their mouths. They look nothing like Hallucigenia except for those claws, the genetic signature that connects them across 500 million years of evolution. The spines on Hallucigenia's back likely defended against Anomalocaris and other Cambrian predators. Anyone trying to swallow this creature would get a mouthful of needles. The defensive strategy worked well enough that Hallucigenia survived for millions of years in seas packed with hunters. But the real mystery is why such a bizarre body plan developed at all. During the Cambrian explosion, evolution experimented wildly with body designs that would never appear again. 5 eyes, circular mouths, spike-covered backs, leg arrangements that defy modern categories. Most of these experiments failed. The creatures died out, leaving only fossils to mark their brief existence. Hallucigenia represents one of the lucky ones, a design strange enough to survive by being too difficult to eat and adaptable enough to eventually crawl onto land and become something entirely different. Scientists named it Hallucigenia because studying it felt like hallucinating, like trying to make sense of an animal that broke every rule of anatomy they understood. 30 years of being wrong about which way was up should have been embarrassing. Instead, it became a reminder of how alien early life truly was, how different the Cambrian seas looked from anything we know today. And Hallucigenia was just the beginning of the strangeness. Imagine an animal 2 inches long with 5 eyes arranged across its head, and a flexible trunk extending from its face like an elephant's, except the trunk ends in a claw. When scientists first presented Opabinia to the Paleontological Association in 1972, the audience laughed. They thought it was a joke, a prank, something cobbled together from multiple fossils as a gag. The reconstruction seemed too absurd to be real. 5 eyes made no sense. The trunk defied every known body plan. The segmented body looked vaguely arthropod-like, but arthropods do not have trunks with pincers on the end. The laughter stopped when more fossils confirmed every impossible detail. Opabinia lived during the Cambrian period, swimming through shallow seas, using lateral lobes that rippled along its body like Anomalocaris. But unlike that apex predator, Opabinia was small, delicate, probably feeding on tiny organisms it plucked from the seafloor, with that bizarre clawed trunk. The five eyes remain the most confusing feature. Most animals with multiple eyes arrange them symmetrically. 2 eyes for depth perception, or many simple eyes scattered across the body for basic light detection. Opabinia had 5 complex eyes in a row across the top of its head, like headlights on a very strange car. Scientists still debate what advantage this gave. Perhaps the extra eyes provided an extremely wide field of vision, letting Opabinia watch for predators from almost every angle while feeding. Or maybe the multiple eyes helped judge distance better when using that flexible trunk to grab food. Or possibly it was just an evolutionary experiment that worked well enough to survive but offered no significant advantage, so it never appeared again. That trunk, though, that was definitely specialized equipment. The flexible appendage extended from between the eyes, bending and curling to reach the seafloor. The claw at the end could grasp small prey, or possibly scavenge dead material. Some scientists think Opabinia might have fed on hallucigenia, using the trunk to carefully pluck the spiky creatures from the ocean bottom and flip them over to reach the soft underside between the legs. The feeding strategy would require precision and patience, which the five eyes might have enabled through superior depth perception and peripheral awareness. But here is what truly bothers scientists about Opabinia. Nobody knows what it is. The body plan does not fit into any modern animal group. It shows some arthropod characteristics like the segmented body, but lacks the jointed legs that define arthropods. It might be related to velvet worms or tardigrades, both groups with unusual body plans themselves. Or it might represent another extinct branch of life that tried something completely different and failed. Recent studies suggest Opabinia might belong to a group called Lobopodia, the same category that includes hallucigenia and velvet worms. If true, that would place it in the ancestry of modern invertebrates, a weird cousin that tried the five-eye trunk approach, while other relatives developed more conventional designs. Fossils of Opabinia come almost exclusively from the Burgess Shale in Canada, those same deposits that preserved Anomalocaris and hallucigenia. The exceptional preservation captured details scientists never expected to see. The individual lenses in each eye, the flexible segments of the trunk, even the delicate lobes used for swimming. Without that fossil site, we might never have known Opabinia existed. The Cambrian seas must have looked like an alien planet, filled with body plans evolution tried once and abandoned. 5 eyes scanning the water, while a trunk reached down to pluck food from mud. Spiky worms, marching on tentacle legs. Giant predators with compound eyes the size of oranges. These were not Earth's oceans as we know them. These were laboratories where life tested every possible design to see what worked. Most failed. A few succeeded. But the ocean was about to get much stranger. The shell rises from the seafloor like a spear stabbed into sand, 20 feet tall, perfectly straight, tapering from thick base to narrow point. Inside lives an animal that invented a completely new way to hunt, using geometry and physics instead of speed or strength. Orthocaris was a nautiloid, a relative of modern squid and octopus, but with a radical difference. While most nautiloids coiled their shells into spirals for compact buoyancy, Orthocaris kept its shell straight as a flagpole and used that shape as a weapon. The animal lived in the bottom chamber of the shell, a soft-bodied creature with tentacles and a sharp beak for tearing prey. The chambers above it, separated by walls called septa, were filled with gas and fluid in carefully controlled ratios. By adjusting the amounts, Orthoceras could rise or sink through the water column with precision. But the straight shell allowed something spiral shells could not: vertical hunting. Orthoceras could hover motionless in the water, pointing straight up like a missile ready to launch. From below, the narrow top of the shell would look like a small fish or piece of debris, easy to ignore. Prey swimming overhead would not recognize the danger until tentacles exploded upward, wrapping around them and dragging them down to the waiting beak. The strategy worked because of how vision functions underwater. Looking down into murky depths, most creatures see vague shadows and dark shapes. Looking up toward light, they see silhouettes clearly outlined against the brightness above. Orthoceras attacked from the blind spot, from the darkness below, where its straight shell blended into the vertical columns of light, filtering through the water. Scientists found evidence of this hunting style in the wear patterns on fossil shells and the types of prey fragments discovered near Orthoceras remains. These nautiloids preferred hunting in areas with vertical structure, near underwater cliffs or drop-offs, where they could position themselves in the shadows and strike upward at passing fish. Some species of Orthoceras grew even longer than 20 feet, reaching lengths that made them the tallest animals in their ecosystems. Imagine swimming in Ordovician seas and passing over what looks like a grove of pale stone pillars rising from the bottom, each one actually a living predator waiting for you to come within reach. The shells themselves were architectural marvels. Each septum that divided the chambers was curved and reinforced to handle water pressure. The sipuncle, a thin tube of tissue running through all the chambers, pumped fluid in and out to control buoyancy. The entire structure had to be strong enough to prevent collapse under its own weight while remaining light enough to float. Orthoceras lived for millions of years, dominating Ordovician and Silurian seas as one of the most successful predator designs. Fossils have been found on every continent, often in such abundance that entire limestone deposits consist almost entirely of Orthoceras shells, packed together like fallen logs. Then, something changed. By the Devonian period, most straight-shelled nautiloids had gone extinct, replaced by coiled ammonites and more advanced fish predators. The vertical hunting strategy that worked so well in earlier seas could not compete with faster, more maneuverable hunters that evolved jaws and proper fins. But for over 100 million years, Orthoceras proved that sometimes the best weapon is not strength or speed. Sometimes the best weapon is geometry, using your shape itself to disappear into the environment and strike from an angle your prey never learned to defend against. The fossils still look like spears, appropriate for an animal that hunted like a thrown javelin. The shell stretched 30 feet from tip to base, wider than a telephone pole, heavy as a small car. Inside lived the largest predator the ocean had ever known at that point in history. Anomalocaris made Anomalocaris look like bait fish. This was a nautiloid that could hunt the hunters, powerful enough to attack any creature sharing Ordovician seas with it. The tentacles alone could wrap around prey the size of modern dolphins. Anomalocaris belonged to the same family as Orthoceras, sharing the straight shell design, but scaled up to nightmarish proportions. The sipuncle running through its shell chambers was thick as your arm, pumping fluids constantly to control buoyancy in a body weighing several tons. The beak at the center of those tentacles could crack through armor that would stop most other predators. Scientists believe Anomalocaris hunted horizontally, not vertically like its smaller relative. The shell was simply too large to maintain the perfect vertical stance Orthoceras used for ambush. Instead, Anomalocaris likely cruised through open water like a modern shark, using jet propulsion by forcing water through a funnel near its head. The massive shell would have made it slow by modern standards, but in Ordovician seas where most prey had limited speed, Anomalocaris was fast enough. The question that haunted scientists for decades was simple but critical. How did something this heavy float? Early reconstructions showed Anomalocaris as a seafloor crawler, too heavy to swim. That changed when researchers calculated the buoyancy provided by the gas-filled chambers. If Anomalocaris maintained the right ratio of gas to fluid in those chambers, the shell would be nearly neutrally buoyant, requiring minimal effort to hover or swim through the water column. But maintaining that balance required constant adjustment. Too much gas and Camaraceras would rise uncontrollably towards the surface, where pressure changes could damage the shell. Too much fluid, and it would sink, potentially getting stuck on the bottom with no way to rise again. The sipuncle had to work continuously, pumping and adjusting, keeping the massive animal suspended at just the right depth. Imagine the energy cost of that system. Camaraceras had to hunt constantly just to fuel the biological pumps keeping it afloat. Any injury to the sipuncle would be fatal, turning the predator into a sinking corpse. Despite the challenges, Camaraceras thrived for millions of years as the apex predator of its time. Fossils show it lived in relatively shallow seas, areas rich with prey fish, smaller nautiloids, and early armored fish. The tentacles could grasp multiple prey items at once, the beak strong enough to crack through the thickest shells available at the time. Then, the Devonian period arrived with its arms race of jaws and armor. Placoderm fish evolved with guillotine-like jaw plates and heavy armor that Camaraceras could not crack. Faster swimmers with better fins could outrun the massive nautiloid. The ecosystem shifted towards speed and agility over raw size and power. Camaraceras went extinct, along with most other giant straight-shelled nautiloids, replaced by smaller coiled ammonites that could maneuver better and required less energy to maintain buoyancy. But for tens of millions of years, Anomalocaris was the ocean's undisputed king. A 30-foot hunting machine with tentacles that could grapple prey larger than most animals alive today. The kind of predator that makes you grateful you were born 450 million years too late to swim in the same waters. The fossils still impress. Shells so large museums display them standing upright like columns holding up ancient temples, which is fitting, because Camaraceras was worshipped by nothing but feared by everything. And what came next made it look small. The claw emerges from murky water, serrated like a bread knife, longer than your forearm. Behind it comes a segmented body covered in chitinous plates, 6 walking legs gripping the riverbed, and 2 paddle-like appendages rowing through the shallows. 8 feet from head to tail spine, this is Pterygotus, the largest arthropod that ever existed. Sea scorpions, called eurypterids by scientists, ruled brackish waters for 200 million years. They were not true scorpions despite the name, more closely related to modern horseshoe crabs and spiders. But the comparison fits when you look at those claws, at the segmented tail that some species could curl over their backs, at the armored body built for hunting in waters where most animals still had soft shells and weak defenses. Pterygotus hunted like an ambush predator, burying itself partially in sand or mud with only its eyes exposed. Those compound eyes on stalks that could rotate independently to scan for movement. When prey swam close, the claws shot forward with enough force to impale fish and drag them back to mandibles that could shred flesh efficiently. But here is what makes sea scorpions truly terrifying. They could breathe air. Not well, not for long periods, but well enough to crawl out of water and cross land between pools. Some species had book lungs, the same breathing system spiders use, allowing them to survive brief periods out of water. Others relied purely on their armored exoskeleton to retain moisture long enough to make short overland journeys. Imagine being a Devonian fish, evolving to escape aquatic predators by developing the ability to breathe air and survive in shallow pools. Then a sea scorpion follows you onto land, claws clacking against stone, hunting you in an environment you thought was safe. Nowhere was safe from these creatures. The largest species, Jacalopterus, reached lengths of over 8 feet and hunted in estuaries where fresh water met salt water. The mixture created rich feeding grounds packed with fish migrating between environments. Jacalopterus would have waded in the murky transition zones, striking at salmon-sized prey swimming past. Scientists found fossilized trackways showing sea scorpions walking on lake bottoms, the marks of their claws visible in stone that was mud 400 million years ago. Other fossils preserve molted exoskeletons, showing these arthropods grew by shedding their armor and expanding before the new shell hardened. During molting, sea scorpions would have been vulnerable, soft-bodied and defenseless for hours or days. They likely hid in crevices or deep water during this period, waiting for the new armor to harden before resuming their role as apex predators. The variety of sea scorpion species was staggering. Some were tiny, barely an inch long, feeding on microscopic prey in shallow pools. Others grew massive, developing specialized claws for different hunting strategies. Some claws were broad and crushing, designed to crack shells. Others were slender and piercing, built to impale fish. Still others had serrated edges for sawing through tough tissue. The evolutionary experimentation produced dozens of body plans, each adapted to specific prey and environments. But the real innovation was the combination of aquatic hunting and terrestrial capability. Sea scorpions pioneered the invasion of land, showing that arthropods could survive and hunt in both environments. This paved the way for scorpions, spiders, and insects to eventually colonize dry land completely. Without eurypterids testing the limits of what chitinous armor could do, the terrestrial invasion might have taken millions of years longer. Then, the Permian extinction arrived and erased 96% of ocean species. Sea scorpions vanished entirely, unable to survive the environmental collapse that ended the Paleozoic Era. Their reign as top predators ended, replaced by marine reptiles and sharks that would dominate the coming Mesozoic seas. But for 200 million years, these armored hunters proved that size, strength, and versatility could make you king of both water and the edges of land. The fossils still look like weapons made animate, because that is exactly what they were. The jaws snap shut with 11,000 pounds of force per square inch, 4 times the bite strength of a great white shark. But these are not teeth. These are razor-sharp bone plates growing from the skull, self-sharpening edges that never dull, never break, never stop growing throughout the creature's life. Dunkleosteus was an armored fish that weighed 4 tons and hunted Devonian seas as the most powerful predator of its time. The head alone measured 3 feet across, encased in thick, bony plates that could deflect attacks from other predators. The plates interlocked like medieval armor, protecting the brain, gills, and vital organs from damage. Behind the armored head, the body was mostly cartilage and muscle, flexible and powerful, built for sudden bursts of speed. But those jaws were the real weapon. Instead of teeth that could break or fall out, Dunkleosteus evolved bone plates that grew continuously from the jaw edges. As the fish bit down on prey, the upper and lower plates ground against each other, keeping both edges sharp as surgical blades. The force of each bite could shear through bone, shell, and thick armor with ease. Scientists calculated the bite force by studying the jaw muscle attachment points and the mechanical advantage of the jaw hinge. 11,000 pounds per square inch is enough to cut a modern human in half with 1 bite. The bite speed was equally terrifying. The jaws closing in 1/50th of a second, faster than most prey could react. Dunkleosteus hunted everything. Fossilized stomach contents show fragments of other armored fish, sharks, and even other Dunkleosteus. This was a cannibal predator that would eat its own species if given the opportunity. The partially digested remains suggest Dunkleosteus could not chew, instead biting prey into chunks and swallowing the pieces whole. But here is where the design shows its flaw. Dunkleosteus had no actual teeth to hold prey, only those blade-like bone plates. Once it bit something, the prey could slip out if it struggled violently enough. To compensate, Dunkleosteus likely used a suction feeding strategy, opening its massive jaws quickly to create negative pressure that pulled prey into its mouth, then slamming the jaws shut before the prey could escape. The combination of suction and slicing made Dunkleosteus nearly unstoppable. Armor that protected other fish from predators was no defense here. The bone plates could shear through even the thickest placoderm armor. Speed offered little help, either because Dunkleosteus could accelerate rapidly for short distances. catching prey before they could reach open water. The only real defense was size, being too large for Dunkleosteus to swallow. Fossils show Dunkleosteus grew throughout its life, with the largest specimens reaching lengths of 33 feet, and possibly more. Some paleontologists argue fragmentary fossils suggest individuals over 40 feet long existed, making Dunkleosteus comparable in size to modern great white sharks, but with far more powerful jaws. The fish lived in shallow seas and estuaries, areas packed with prey fish and other placoderms. Competition between Dunkleosteus individuals must have been fierce, with the largest fish dominating the best hunting grounds and smaller individuals forced into marginal areas. Evidence of healed bite marks on Dunkleosteus fossils confirms they fought each other regularly, probably over territory or mates. But the armor that made Dunkleosteus nearly invincible also limited it. The heavy headplates required constant swimming to maintain depth, as the fish had no swim bladder to control buoyancy. This burned enormous amounts of energy, forcing Dunkleosteus to hunt frequently just to fuel its metabolism. Any injury that prevented swimming would be fatal, the armored fish sinking to the bottom where it would suffocate. Dunkleosteus went extinct at the end of the Devonian period, along with most other placoderms. The reasons remain debated. Some scientists blame environmental changes that reduced prey populations. Others point to competition from early sharks that were faster and more efficient. The most likely explanation combines both factors, along with the Kellwasser extinction events that killed off many marine species. But for tens of millions of years, Dunkleosteus was proof that bone could be deadlier than teeth, that armor could turn a fish into a swimming tank, that raw power still ruled even as evolution favored speed. The fossils still show those blade edges, still sharp after 350 million years, still looking hungry. The spiral of teeth set in a drawer at the American Museum of Natural History for decades, labeled as an ammonite shell. It was not a shell, it was a mouth. And scientists had no idea which end of the shark it belonged to. Helicoprion was a prehistoric shark that lived from the Permian through the Triassic periods, distinguished by a bizarre tooth spiral that looked like a circular saw blade. Early reconstructions placed the whorl on the shark's nose like a battering ram, on its dorsal fin like a weapon, even on its tail like a defensive spike. They were all catastrophically wrong. The tooth whorl grew from the lower jaw, curving backward into the shark's throat in a continuous spiral that added new teeth throughout the animal's life. Older teeth rotated inward as newer teeth grew at the front, creating a buzzsaw structure that never stopped growing and never wore down completely. But why would evolution create such an absurd feeding mechanism? The answer lies in what Helicoprion ate. The shark specialized in hunting soft-bodied prey like squid, cuttlefish, and ammonoids, all creatures with hard beaks but relatively soft bodies. The tooth spiral worked like a conveyor belt, dragging prey into the mouth while the curved teeth prevented escape. As the shark bit down, the spiral rotated slightly, pulling the prey deeper into the throat where it could be swallowed whole. Scientists finally understood the mechanism when CT scans of well-preserved fossils revealed how the whorl connected to the jaw joint. The spiral sat inside the lower jaw, protected by cartilage and muscle tissue that is not fossilized. Only the teeth survived, leaving earlier paleontologists to guess at their placement. Some Helicoprion whorls measure 3 feet in diameter, suggesting individuals over 25 feet long. These were not small sharks. They hunted in open ocean, chasing down ammonoids and other cephalopods in waters from shallow shelves to deep basins. The tooth count in some whorls exceeds 180 individual teeth, each one serrated and curved like a scimitar. The growth pattern tells us Helicoprion lived for decades at minimum, possibly much longer. Sharks in general grow slowly and live long lives, and Helicoprion's tooth spiral shows growth bands similar to tree rings, indicating periods of fast and slow growth, corresponding to seasonal changes in food availability. But here is what scientists still cannot answer. How did the shark swallow food with that spiral in the way? The whorl curved backward into the throat, which should have blocked anything Helicoprion tried to eat. Current theory suggests the shark had an extremely flexible throat that could expand around the whorl, or possibly the prey was crushed and torn by the teeth before being swallowed in pieces. The exact mechanism remains a mystery because soft tissue does not fossilize. What we know for certain is that Helicoprion's design worked successfully for over 150 million years, surviving multiple extinction events that killed off most other marine species. The buzzsaw jaw gave it a specialized, niche-hunting soft-bodied cephalopods, prey that other sharks struggled to catch and hold. Then, the end Permian extinction arrived and erased 96% of ocean life. Helicoprion survived that event, one of the few large predators to make it through, and continued hunting in Triassic seas for another 40 million years. But competition from more advanced sharks and marine reptiles eventually pushed Helicoprion to extinction. The tooth whorls still confuse people when they see them in museums. Is that a fossil shell? They ask. No, that is a mouth. A buzzsaw mouth that spun prey into a shark's throat for 150 million years. Proof that evolution does not care about elegant solutions as long as the solution works. And sometimes the solution looks like power tools. The fossil shows an animal 8 inches long with a trunk extending from its head, ending in a claw. Sound familiar? Except, this trunk is much longer than Opabinia's, stretching half the body, length before terminating in that grasping appendage. The eyes sit on stalks protruding from opposite sides of the body, set far apart like a hammerhead shark's. The body itself appears soft, lacking any obvious segmentation or hard structures. This is Tullimonstrum gregarium, commonly called the Tully monster, and scientists have been arguing about what it is for over 60 years. Is it a mollusk, an arthropod, a worm, a vertebrate? Something else entirely? The Tully monster was discovered in coal deposits in Illinois, preserved in such abundance that it became the official state fossil. Thousands of specimens have been collected, studied, photographed, and analyzed with every technology available. We have Tully monster fossils in every size from juveniles to adults, showing growth patterns and variation between individuals, and we still cannot agree on what it is. The trunk suggests mollusk, possibly related to squid or snails, but mollusks do not have eye stalks positioned like that. The eye placement suggests vertebrate, similar to how fish position their eyes. But vertebrates do not have trunks with claws. The soft body and lack of shell suggest worm, but worms do not have eyes on stalks or grasping appendages. Every feature points to a different animal group, making classification impossible. Recent studies using advanced imaging techniques claim to have found evidence of a notochord, the flexible rod that defines vertebrates. If true, this would make Tully monster a bizarre fish-like creature, possibly related to lampreys. The study caused controversy because other scientists could not replicate the findings, and accused the researchers of seeing patterns in artifacts of preservation rather than actual anatomical structures. The debate continues with no resolution in sight. What everyone agrees on is how the Tully monster fed. The trunk was flexible and mobile, reaching out to grab small prey or scavenge dead material from the seafloor. The claw at the end had tiny teeth-like structures that could grip slippery food. The eyes on stalks provided wide-angle vision, letting the Tully monster watch for predators while feeding. But why position the eyes so far apart? The eye stalks stretch almost to the edges of the body, giving the Tully monster the widest eye span relative to body size of any known animal. This arrangement creates excellent depth perception and peripheral vision, useful for an animal feeding in murky water where visibility is limited, and predators could approach from any angle. The Tully monster lived in shallow marine environments, areas filled with soft mud and decaying vegetation. The ecosystem it inhabited was essentially a coastal swamp, warm and productive, packed with life. Fossils show the Tully monster was itself prey for larger predators. Some specimens have bite marks or are preserved inside the guts of fish, proving these strange creatures were part of the food chain despite their alien appearance. The abundance of Tully monster fossils is itself a mystery. Why did so many of these animals die and get preserved in one relatively small area? Most likely the environment had low oxygen levels at the bottom, preventing scavengers from eating the dead Tully monsters before they could fossilize. The bodies sank into soft mud that excluded oxygen, preserving even the soft tissue as impressions in stone. But this does not explain why they were so common in the first place. Did Tully monsters congregate for breeding? Were they migratory animals that traveled through this area seasonally? Or were they simply so successful in this environment that population density reached levels that made fossilization common? Nobody knows. What we have are thousands of fossils of an animal that might not even be an animal in the traditional sense. A creature so bizarre, it defies every attempt to place it in the tree of life. The Tully monster lived. The Tully monster hunted. The Tully monster bred and died, and left behind evidence of its existence. We just cannot figure out what the hell it was. And honestly, that might be the most important lesson the Tully monster teaches us about Paleozoic seas. Some things were so alien, we will never fully understand them. The eye is built from calcite, the same transparent mineral that forms limestone. Not an eye socket holding a soft lens. Not an eye protected by transparent tissue. An actual eye made of rock, grown atom by atom into a precise optical structure that could focus light, track movement, and provide vision clearer than what most modern arthropods possess. Trilobites invented crystalline eyes 500 million years ago and used them to dominate ocean floors for 270 million years. The design was engineering perfection. Each eye contained hundreds of individual lenses, all made from pure calcite crystals oriented in specific directions to minimize distortion. The lenses were hexagonal, fitting together like bathroom tiles with no gaps between them. Light passing through one lens would be focused onto a specific point on the retina beneath, creating a mosaic image made of hundreds of individual views. But calcite has a problem. The mineral is birefringent, meaning it splits light into two rays traveling at different speeds. This would normally create double images, making the eye useless. Trilobites solved this with physics. They grew the calcite crystals with their optical axis perfectly aligned with the incoming light, cancelling the birefringent effect. Each lens was a single crystal oriented precisely to let light pass through without splitting. The mathematical precision required to grow eyes this way is staggering, implying trilobites had genetic control over crystal formation at the atomic level. Some species went further, developing compound lenses made of two calcite crystals bonded together in an arrangement that corrects spherical aberration, the distortion that makes curved lenses blur images at the edges. This two-crystal lens design would not be reinvented by humans until the 17th century, when mathematician Christian Huygens and philosopher René Descartes independently derived the mathematics. Trilobites beat them to it by 500 million years. The eyes gave trilobites incredible advantages. They could see predators approaching from almost any angle. Some species had eyes on stalks that could rotate independently, providing 360-degree vision. Others had eyes positioned to look upward, watching for threats from above while feeding on the seafloor. Different species evolved different eye strategies. Bathymetric trilobites living in deep water had enormous eyes with huge lenses optimized for gathering minimal light. Shallow water species had smaller eyes with more lenses, trading light sensitivity for higher resolution. Some trilobites that lived in caves or very deep trenches lost their eyes entirely, becoming blind and relying on other senses to navigate and feed. But the crystal eyes had vulnerabilities. Unlike soft tissue eyes that can heal from minor damage, calcite lenses could not repair themselves. A crack or chip would permanently impair vision. Trilobites molted their exoskeletons regularly as they grew, and each molting included shedding the eye lenses and growing new ones, an opportunity to replace damaged lenses with fresh ones. The fossil record preserves these molted eyes in perfect detail, thousands of individual lenses frozen in stone, still clear enough that scientists can test their optical properties by shining light through them. Some fossilized trilobite eyes still focus light after 500 million years. The preservation is possible because the eyes were already rock when the animal was alive. Calcite fossilizes easily because it is already mineral. Where soft tissue eyes leave only impressions or decay completely, crystal eyes remain as functional optical instruments, proof of how well the design worked. Scientists have identified over 20,000 trilobite species, each with variations on the crystal eye design. Some had eyes as large as ping pong balls, others had eyes barely visible without magnification. A few species had eyes covering most of their head, creating nearly spherical vision with minimal blind spots. The diversity proves eyes were crucial to trilobite survival. In seas packed with predators like Anomalocaris, Anomalocaris, and eventually Dunkleosteus, vision meant the difference between escaping and becoming food. Crystal eyes gave trilobites that edge, the ability to spot danger early and react before the predator could strike. Then, the Permian extinction erased them completely. Every single trilobite species went extinct, ending a lineage that had survived 4 previous mass extinctions. The crystal eyes that served them for 270 million years could not save them from environmental collapse. But the eyes remain, locked in stone, still functional, still focusing light on no retina, still watching empty seas. The trilobites are gone. Their eyes still see. The creature measures 1 inch long, transparent as jellyfish, barely visible in clouded Cambrian waters. No jaws, no proper fins, no bony skeleton, just a flexible rod running from head to tail, small gill slits along the body, and primitive eyes that can barely distinguish light from dark. This is Hycoichthys, and every vertebrate alive today descended from it. You have a spine because this animal had a notochord, that flexible rod made of stiffened tissue that would eventually become your backbone. You have a skull because Hycoichthys had a primitive head structure protecting a simple brain. You breathe with lungs descended from the gill slits this creature used to filter oxygen from water. The entire vertebrate family tree, from fish to humans, traces back to something smaller than your thumb that swam in seas 520 million years ago. Hycoichthys was not the first animal with a notochord. Other creatures like Pikaia had similar structures, but Hycoichthys shows features that mark it as the earliest known craniate, the group that includes all animals with skulls. The head had cartilage structures protecting the brain and supporting the gills, a crucial innovation that allowed more complex nervous systems to develop. The animal swam like an eel, flexing its body in S-shaped waves to push through water, no muscular tail for powerful swimming, no paired fins for stability or steering, just simple undulation, the most basic form of aquatic locomotion. This made Hycoichthys slow and vulnerable, easy prey for the predators that ruled Cambrian seas. Survival required hiding, staying near the seafloor, feeding on tiny organisms that larger predators ignored. The fossil record shows Hycoichthys had a filter feeding system, using its gill slits to strain plankton and organic particles from water. The mouth had no jaws, just an opening that sucked in water and expelled it through the gills, trapping food particles on specialized structures inside. This jawless design limited what Hycoichthys could eat, restricting it to the smallest prey items. But the limitations created opportunity. While armored predators like Anomalocaris fought over larger prey, Hycoichthys exploited a niche they could not access. Microscopic plankton blooms provided unlimited food for filter feeders. No competition from creatures too large or too specialized to bother with such tiny meals. The strategy worked for millions of years. allowing early vertebrates to establish themselves and eventually evolve more sophisticated features. The notochord was the key innovation. That flexible rod provided attachment points for muscles stronger than what soft-bodied animals could develop. More muscle power meant faster swimming, better escape responses, improved hunting ability. The notochord would eventually ossify into bone, creating the vertebral column that defines vertebrates. Hycoichthys fossils come from the Chengjiang deposits in China, the same formation that preserved Anomalocaris and countless other Cambrian creatures. The preservation captured details scientists never expected to see in animals this ancient. The outline of the notochord appears as a dark line. The gill slits show as openings along the body. Even the eye structures remain visible as small dark spots. Without this exceptional preservation, we might never have found evidence of our earliest vertebrate ancestor. The transition from Hycoichthys to modern vertebrates required hundreds of millions of years and countless evolutionary steps. Jaws evolved, fundamentally changing what vertebrates could eat and how they hunted. Paired fins appeared, enabling precise maneuvering and stability. Bony skeletons replaced cartilage, providing support for larger body sizes and stronger muscles. But it all started with a 1-inch filter feeder that barely qualified as a predator. A creature so primitive, it makes modern fish look like technological marvels. You exist because Hycoichthys survived. Your spine, your skull, your nervous system, all refined versions of structures this tiny animal pioneered in Cambrian seas. Every breath you take uses a system descended from those primitive gill slits. Every thought processes through a brain housed in a skull derived from the cartilage structures protecting Hycoichthys' simple neural cluster. 520 million years of evolution separate you from this ancestor, but the connection is direct and undeniable. Somewhere in your genome, encoded in DNA passed down through countless generations, are instructions originally written in Hycoichthys, refined and modified, but never entirely erased. The thumb-sized filter feeder never knew it was founding a lineage that would eventually dominate every environment on Earth. It just kept swimming, filtering plankton, and surviving long enough to reproduce. Sometimes that is all evolution requires. It looks like a worm with racing stripes. 2 inches long, semi-transparent, with V-shaped muscle segments visible through its skin like chevrons pointing toward the head. A small tentacled structure protrudes from the head area, possibly for feeding or sensing the environment. Pikaia is the earliest known chordate, the first animal with a notochord that clearly belongs in our evolutionary lineage. The notochord runs the length of the body, a flexible rod that provides support and attachment points for muscles. This is the structure that would eventually become the spine, transforming from a simple rod of stiffened tissue into segmented vertebrae made of bone. Every animal with a backbone traces its ancestry through Pikaia. But Pikaia itself had no bones, no skull, no vertebrae. Just that flexible rod and the muscle segments that pulled against it, flexing the body for swimming. The movement was more efficient than what soft-bodied worms could achieve, giving Pikaia slightly better speed and maneuverability. Not much of an advantage, but enough to make a difference in Cambrian seas, where small improvements in escape speed meant survival. Scientists found Pikaia fossils in the Burgess Shale, the same formation that yielded Opabinia, Hallucigenia, and Anomalocaris. For decades after its discovery, researchers debated what Pikaia was. The tentacled head structure looked unlike anything modern. The muscle pattern suggested chordate, but the evidence was not conclusive. Then more fossils emerged, showing the notochord clearly, along with other chordate features. The debate shifted from whether Pikaia was a chordate to how it fit into chordate evolution. Was it a direct ancestor of vertebrates, or a side branch that left no descendants? Did it represent the transitional form between simpler organisms and the first fish, Or was it already too specialized to be a true ancestor? Current consensus places Pikaia as a very early chordate, possibly not directly ancestral to vertebrates, but close enough to show what the common ancestor might have looked like. The tentacles remain mysterious, possibly used for filter feeding like Hycoichthys, or for sensing chemical cues in the water. What made Pikaia significant was not just the notochord, but the entire body plan. The bilateral symmetry with distinct head and tail ends. The segmented muscles allowing wave-like swimming motions. The simple nerve cord running along the notochord, a prototype of the spinal cord that would later develop in vertebrates. These features combined to create a blueprint for success that evolution would refine for the next 500 million years. Pikaia was not a predator. The small size and lack of obvious feeding structures suggest it filtered plankton or grazed on bacterial mats. This put it in direct competition with countless other filter feeders, all competing for the same microscopic food sources. The notochord gave Pikaia enough of an edge to survive and reproduce. That edge was everything. If Pikaia had gone extinct, if some random event had killed off the population before it could diversify, vertebrate evolution might have taken a completely different path. Other chordates existed in Cambrian seas, but Pikaia appears to be the one closest to our own lineage. Its survival was not inevitable. Fossils are rare, suggesting low population numbers or limited range. One bad storm, one predator finding the population, one environmental shift could have ended the lineage before it truly began. But Pikaia persisted. The muscle patterns visible in fossils show the same basic arrangement modern fish use for swimming. The V-shaped segments, called myomeres, pull against the notochord in alternating waves, creating the side-to-side body movement that propels fish through water. This design is so efficient that evolution kept it for 500 million years with only minor modifications. You can see the same pattern in tuna, sharks, eels, even in the embryonic development of land vertebrates before their bodies adapt to terrestrial life. Pikaia pioneered that design when the ocean was still figuring out basic body plans. The tentacles on the head likely had sensory functions, detecting chemicals dissolved in water to find food or avoid predators. Modern fish retain similar structures in their barbels and taste buds, organs that let them taste the water around them and track prey by chemical signature alone. 520 million years ago, Pikaia swam through Cambrian seas, looking like a 2-inch worm with racing stripes, filtering plankton and avoiding predators. It had no way of knowing it was carrying the blueprint for fish, amphibians, reptiles, birds, and mammals. It just kept swimming, kept feeding, kept reproducing, and accidentally founded the most successful vertebrate lineage in Earth's history. Sometimes, the most important innovations look insignificant at first. A worm with a stiffening rod. That is all it was. That is all it needed to be. The world changed the moment the first mouth closed around prey and held it. Before jaws, predators were limited to grasping appendages like Anomalocaris, or suction feeding like early fish. Prey could slip away if they struggled. Armored shells offered protection from most attacks. The ocean was still a relatively safe place if you had hard covering and could avoid the largest predators. Then placoderms evolved jaws, and the arms race exploded. Jaws were hinged structures made of modified gill arches, the bones that originally supported breathing apparatus. Evolution repurposed them, adding powerful muscles and sharp edges, creating a mechanism that could bite down and hold with crushing force. The first jaws were simple, basically bone shears that opened and closed, but simple was enough to revolutionize predation. Placoderms could now grip prey firmly, preventing escape. They could bite through shells that had protected animals for millions of years. They could tear chunks from larger prey instead of needing to swallow everything whole. The feeding efficiency increased dramatically, allowing placoderms to exploit food sources other predators could not access. Within millions of years, hundreds of placoderm species evolved, each with specialized jaw modifications. Some developed long-pointed jaws for catching fish. Others evolved broad crushing jaws for eating shellfish. Still others created slicing jaws with blade-like edges for cutting through armored prey. The diversity exploded as jaws enabled niche specialization on a scale previously impossible. Dunkleosteus represented the peak of placoderm evolution, jaws refined into guillotine blades with enough force to shear through bone. But many other placoderms were equally bizarre in their own ways. Bothriorlepis had jaws but barely used them, instead developing armored pectoral fins that looked like crab claws. These fish spent most of their time on the seafloor, using the modified fins to pull themselves through mud while sucking up organic material. The jaws were small and weak, used only for processing the softest food. Titanichthys grew to 25 feet long, but had jaws too small and weak to be effective predators. Scientists believe this giant placoderm was a filter feeder despite belonging to the same family as Dunkleosteus. The jaws might have served some other function, perhaps social displays or territorial disputes, while the fish fed by swimming with its mouth open and straining plankton. Philolepids flattened themselves like pancakes, developing armor so heavy and bodies so broad they could barely swim. They lived on river bottoms in freshwater environments, using their jaws to grind plant material and detritus. The armor protected them from the enormous predators that shared their habitats. But jaws created a new problem. The more powerful the jaw muscles became, the larger and heavier the head had to be to anchor those muscles and support the forces involved in biting. Placoderms compensated by developing heavy armor plates covering the head and front section of the body. This made them front-heavy and slow, requiring constant swimming to avoid sinking. The energy cost was enormous, forcing placoderms to hunt frequently just to fuel their metabolisms. Meanwhile, another group was evolving a better solution. Sharks developed jaws without heavy armor, using cartilage instead of bone to reduce weight. Their streamlined bodies and efficient swimming made them faster and more energy efficient than armored placoderms. When the Devonian period ended, environmental changes favored speed and efficiency over size and armor. Placoderms went extinct, every single species vanishing within a few million years. Sharks survived and diversified, taking over many of the ecological roles placoderms had filled. The age of armored fish ended, replaced by the age of sleek predators. But placoderms had already changed the ocean forever. By inventing jaws, they started an evolutionary arms race that continues today. Prey evolved thicker shells, faster swimming, better camouflage, and chemical defenses. Predators responded with stronger jaws, faster pursuit, improved senses, and pack hunting strategies. Each innovation drove counter-innovations, pushing both predators and prey to evolve at accelerating rates. The Cambrian explosion had created diverse body plans and basic predation strategies. The Devonian jaws race refined those strategies into the sophisticated predator-prey dynamics we see in modern oceans. Without placoderms, fish might never have developed the feeding versatility that allowed them to dominate aquatic environments. Sharks might never have evolved beyond simple scavengers. The entire trajectory of vertebrate evolution would have been different, all because some ancient fish repurposed breathing structures into weapons. The fossils still show those jaws, still open as if ready to bite, still demonstrating the innovation that transformed the ocean from a garden into a battlefield. Jaws changed everything, and placoderms were the ones who opened that door. The shells packed together like cobblestones, thousands of individuals clustered so tightly that walking across the reef would have felt like walking on pavement. These were not corals building the reef. These were brachiopods, clam-like animals with two shells that built massive colonies covering square miles of seafloor. For 300 million years, brachiopods dominated shallow oceans as the most successful, shelled organisms on Earth. They looked similar to clams, but were completely different animals. Clams are mollusks, related to snails and squid. Brachiopods belong to their own unique phylum, more closely related to bractozoans and phoronids than to anything with a shell. The similarity is convergent, evolution, two unrelated groups independently developing hinged shells as protection from predators. But brachiopod shells open differently than clamshells. Clamshells open sideways, left and right. Brachiopod shells open vertically, top and bottom, with the line of symmetry running through the shells rather than between them. This seems like a minor difference, but reflects completely different internal anatomy and feeding strategies. Brachiopods fed using a lopophore, a specialized feeding structure covered in tiny hair-like cilia that created water currents. The currents pulled plankton and organic particles into the shell, where the lopophore filtered out food and expelled the filtered water. The system was incredibly efficient, allowing brachiopods to thrive in environments with relatively low nutrient levels. During the Paleozoic era, brachiopods built reefs that rivaled modern coral reefs in size and complexity. The shells cemented together as individuals settled on top of previous generations, creating vertical structures that rose dozens of feet above the seafloor. These reef cities provided habitat for countless other species. Fish sheltered between the shells. Small arthropods grazed on algae growing on the shell surfaces. Predators patrolled the reef edges, hunting the abundant prey. Brachiopod reefs were the foundation of entire ecosystems. Some species developed elaborate shell ornamentation. Spines protruded from the shell edges, possibly for defense or to prevent sinking into soft sediment. Ridges and grooves created patterns that might have helped strengthen the shell or increase surface area for the lopophore. Colors varied from white to red to gold, though most fossil specimens have lost the original pigmentation. The diversity of brachiopod species was staggering. Scientists have identified over 12,000 fossil species, each with unique shell shapes and feeding strategies. Some were smaller than a fingernail, others grew larger than your fist. A few species developed asymmetric shells, with one side much larger than the other for reasons still debated. Competition between brachiopods and clams had been ongoing since both groups appeared. During the Paleozoic, brachiopods dominated completely. They outnumbered clams by huge margins in nearly every marine environment. The lopophore feeding system seemed superior to the clam's gill-based filter feeding. Brachiopod shells were generally stronger and more resistant to predator attacks. But the Permian extinction changed everything. 96% of ocean species went extinct, including the vast majority of brachiopod species. The reef-building forms died out completely. Only a few generalist species survived in deep water refuges or isolated environments where the extinction effects were less severe. Clams somehow weathered the extinction better. They diversified rapidly in the aftermath. Filling niches brachiopods had dominated for 300 million years. By the time the Triassic period began, clams outnumbered brachiopods and have maintained that dominance ever since. Scientists still debate why clams survived better than brachiopods. Some theories focus on reproduction, with clams able to produce more offspring faster than brachiopods. Others point to feeding efficiency, suggesting gill-based filter feeding works better in the low oxygen conditions that followed the extinction. Still, others argue it was simply luck, with the specific environmental changes hitting brachiopod populations harder than clam populations. Whatever the reason, the shift was permanent. Today, only about 400 brachiopod species exist, mostly in deep water or specialized environments. They are considered living fossils, organisms that have changed little over hundreds of millions of years. The shells still open vertically. The lopophore still filters food from water currents. The basic body plan remains unchanged from Paleozoic ancestors, but the dominance is gone, replaced by clams and other mollusks. The brachiopod reef cities are fossil now, limestone deposits that mark where shallow seas once teemed with life. You can still see the individual shells packed together in stone, thousands of lives compressed into layers of rock. Some formations contain so many brachiopod fossils, they are mined as building materials, entire reefs recycled into modern construction. 300 million years of dominance ended in less than 1 million years of environmental collapse. The shells remain as monuments to what once ruled the seafloor, and reminders that dominance is never permanent. For over a century, scientists found the teeth and nothing else. Microscopic structures shaped like combs, fans, blades, and platforms, all made of hard mineral tissue similar to tooth enamel. They appeared in rock formations spanning 300 million years, from the Cambrian to the Triassic periods, in such abundance that geologists use them to date rock layers and correlate formations across continents. But what animal did the teeth belong to? The mystery frustrated paleontologists for generations. The teeth were clearly functional, showing wear patterns that proved they were used during the animal's life. Some had multiple points like combs, others formed platforms with complex surface textures. Still others looked like curved blades designed for slicing. Yet no one could find the body attached to those teeth. Soft-bodied animals rarely fossilize, leaving behind only their hardest parts if they fossilize at all. The conodont teeth were made of apatite, the same mineral in human teeth and bones, making them resistant to decay and easily preserved. The bodies apparently lacked any hard structures and decayed before fossilization could occur. Hundreds of theories emerged over the decades. Maybe conodonts were worms with mineralized mouthparts. Perhaps they were early fish with tooth-like feeding structures. Some suggested they were completely unknown animals with no modern relatives. Others argued they might be parts of larger organisms, serving unknown functions unrelated to feeding. Then, in 1983, a farmer in Scotland found the first complete conodont fossil. The animal was eel-like, 2 inches long, with large eyes and a notochord running the length of its body. The teeth sat in a complex arrangement inside the throat area, forming a feeding apparatus unlike anything in modern animals. Multiple tooth elements worked together like a precision machine, grasping and crushing prey with coordinated movements. Conodonts were chordates, early relatives of vertebrates, using mineralized feeding structures similar to teeth, but arranged in ways no modern animal employs. The discovery solved one mystery, but created others. How did the tooth apparatus function? The elements did not mesh together like human teeth. They seemed to work by moving independently, each element performing a specific role in the feeding process. Some elements might have been used for grasping prey. Others crushed food. Still others cut or strained material. Scientists built mechanical models trying to reconstruct how the system worked, with limited success. The feeding apparatus was simply too different from anything modern to allow confident reconstruction. We can see that it worked because the teeth show wear patterns consistent with heavy use. We just cannot figure out exactly what motions create those wear patterns. Conodont teeth changed throughout their evolutionary history, reflecting changes in diet and feeding strategies. Early species had simple, cone-shaped teeth, probably used for grasping small prey. Later species developed complex platforms and blade-like elements, suggesting more sophisticated processing of food. The most advanced conodonts had tooth arrangements so intricate that reconstructing them requires careful study of how different elements fit together. The variety of tooth forms made conodonts extremely useful for dating rocks. Different species evolved distinct tooth shapes that appeared and went extinct at specific times. Geologists could identify which rock layers were older or younger based on which conodont teeth they contained. This biostratigraphy allowed correlation of formations across huge distances, helping build the geologic timescale we use today. Ironically, conodonts became more useful as geological tools than as understood animals. The teeth were so abundant and well preserved that studying them was easy. The bodies were so rare and poorly preserved that understanding the actual animal remained nearly impossible. Even now, with complete specimens available for study, scientists still debate exactly how conodonts lived, fed, and moved through Paleozoic seas. What we know is they were successful, thriving for 300 million years in environments from shallow reefs to deep ocean basins. They survived multiple mass extinctions that killed off most other marine life. They diversified into hundreds of species, each with unique tooth arrangements adapted to specific diets. Then, the end Triassic extinction erased them completely. Every conodont species went extinct, ending a lineage that had persisted longer than dinosaurs would exist. The teeth that had served them for 300 million years could not save them from whatever environmental disaster triggered the extinction. Now, we find only the teeth, scattered through rock formations like cryptic messages from animals we barely understand. For 100 years, they were a mystery without a solution. Now they are a solution that raises more questions. Sometimes finding answers just reveals how much we still do not know. The fish bristled with spines like an underwater porcupine. 15 spines protruding from a body barely 6 inches long. 2 large spines in front of the pectoral fins. Smaller spines before the pelvic fins. Multiple spines along the belly, between paired fins. 2 dorsal spines on the back. A spine before the anal fin. Even the tail had supporting spines. Climatius was an acanthodian, a group of early fish sometimes called spiny sharks, though they were not true sharks. The spines were the defining feature, bony structures covered in enamel-like tissue that could not retract or fold against the body. Every spine was permanent, locked in place, jutting from the fish at angles that must have made swimming awkward. So why evolve so many spines? The obvious answer is defense, making Climatius too difficult to swallow for most predators. Any creature trying to eat this fish would get a mouthful of sharp bony projections that could damage mouth tissue or get lodged in the throat. The defense was passive, requiring no action from Climatius beyond having the spines in the first place. But defense does not explain why Climatius had more spines than other acanthodians. Most spiny sharks had 4 to 8 spines total, enough for deterrence without excessive weight or drag in the water. Climatius had 15 or more, depending on the species, far beyond what simple predator deterrence required. The extra spines must have served additional functions beyond making the fish unpalatable. Some scientists suggest the spines provided hydrodynamic advantages. Properly arranged, they could create vortices that reduce drag or increase maneuverability. Modern fish use fin spines for similar purposes, creating turbulence that helps control movement at slow speeds. Climatius might have used its excessive spine count to achieve precise control in complex environments like reefs or vegetation beds. Others argue the spines were sexual displays. Males might have had more elaborate spines than females, using them to attract mates or intimidate rivals. Sexual selection could drive the evolution of structures far beyond what survival alone would require. Peacock tails and elk antlers prove that display purposes can create seemingly excessive ornamentation. The problem is we cannot test these theories easily. Fossil fish rarely preserve evidence of sexual dimorphism clearly enough to distinguish male from female specimens. We have no way to know if some Climatius individuals had more ornate spines than others, or if all members of the species looked identical. What we know for certain is the spines worked as defense. Fossil evidence shows Climatius lived alongside major predators like early placoderms and larger acanthodians. Despite being small and probably not fast swimmers, Climatius survived and diversified into multiple species. The spines must have provided enough protection to offset the disadvantages they created. And there were disadvantages. Each spine added weight that required more energy to move through water. The spines created drag, slowing swimming speed. They increased the fish's profile, making it easier for predators to spot. In environments with dense vegetation or narrow crevices, the spines could get caught or tangled, trapping the fish. Climatius paid significant costs for its armor. The spines were supported by internal bones and muscles, requiring resources to build and maintain. Growing new spines as the fish matured demanded calcium and other minerals that could have been used for other purposes. The trade-off suggests the defensive benefits were substantial enough to justify the expenses. Acanthodians like Climatius were among the first fish to develop paired fins and spines, innovations that would later be refined in sharks and bony fish. The spines in front of the fins probably stabilized them, preventing the fins from bending backward under water pressure during swimming. This stabilization allowed more powerful fin strokes, increasing swimming efficiency despite the drag created by the spines themselves. The evolutionary experimentation with spine placement and number eventually settled on more moderate designs in later fish groups. Sharks developed spines only in front of dorsal fins, reducing drag while retaining defensive benefits. Bony fish mostly abandoned spines altogether, relying on scales and speed for protection. But during the Silurian and Devonian periods, Climatius proved that maximum spines could be a viable strategy. The fish survived for tens of millions of years, long enough to leave abundant fossils in formations across North America and Europe. The preservation often shows the spines in perfect detail, each one positioned exactly as it was when the fish died. Looking at those fossils, you cannot help but wonder what swimming through Devonian seas felt like for this mobile pincushion. Every movement would have been calculated to avoid snagging on rocks or plants. Every escape from predators would have been a balance between speed and the risk of tangling the spines. Every interaction with other Climatius would have required care to avoid injuring each other with those sharp projections. Living with 15 permanent spines could not have been comfortable, but it kept Climatius alive in seas that killed most creatures without hesitation. Sometimes, survival requires accepting discomfort. The spines were the price Climatius paid to exist, and for millions of years, that price was worth paying. The shell floated just beneath the surface, pointing downward like a javelin thrown into water. 3 feet long, gently curved, the tip almost invisible from above, while the wide base caught sunlight and created shadows that confused prey about what they were seeing. This was an orthoconic nautiloid, a hunter that used verticality as a weapon in shallow seas, where vertical space offered the only real hunting advantage. These were cousins to the massive Orthoceras and Camaraceras from deeper waters, but evolved for a different environment entirely. Shallow seas during the Ordovician and Silurian periods were crowded with life. Fish schooled in the sunlit waters. Smaller nautiloids hunted along the bottom. Trilobites scuttled through seagrass. The ecosystem was dense, competitive, packed with both prey and predators fighting for the same resources. Vertical hunting gave orthoconic nautiloids an edge others lacked. The straight or gently curved shell allowed them to hover at any depth, adjusting buoyancy to hang motionless in the water column. From this position, they could scan both above and below, watching for prey while staying alert for larger predators. The tentacles emerging from the base of the shell could strike in any direction with little warning, but shallow water meant different challenges than deep water hunting. Light penetrated to the seafloor, eliminating the darkness that deep water orthoceras used for concealment. Currents were stronger, requiring constant adjustment to maintain position. Prey had better vision in bright water, making ambush more difficult. Temperature fluctuations between day and night, between seasons, stressed animals adapted to stable, deep-sea conditions. Shallow water orthoconic nautiloids adapted by developing faster reflexes and more aggressive hunting behavior. They could not rely on ambush alone. Instead, they actively pursued prey, using jet propulsion to accelerate quickly and grab fish before they could escape. The tentacles had stronger muscles than their deep water cousins, capable of holding struggling prey more firmly. The beak was sharper, designed to kill quickly rather than subdue slowly. Some species developed elaborate shell patterns, possibly for camouflage. Color patterns on the shells might have broken up the nautiloid's outline when seen from above, making it harder for prey to recognize the predator. Banding in different shades could have mimicked light filtering through water, helping the nautiloid blend into the background despite its size. We cannot know the original colors because they rarely fossilize, but the patterns preserved in some specimens suggest sophisticated visual deception. The shells themselves show signs of frequent damage and repair. Healed fractures indicate these nautiloids engaged in violent encounters, possibly with prey, possibly with each other, or possibly with predators that attacked but failed to kill them. Each fracture would have compromised buoyancy temporarily, forcing the nautiloid to compensate by adjusting fluid levels in other chambers. Severe damage could be fatal if it prevented the animal from maintaining depth control. Orthoconic nautiloids in shallow waters also had to deal with seasonal changes that deep water species never experienced. Temperature swings affected metabolism, requiring different hunting strategies in warm versus cold water. Storms could physically damage shells or force nautiloids into unsuitable depths. Seasonal migrations of prey meant feast or famine depending on timing. These environmental pressures favored smaller, more adaptable species over the giants that dominated deeper waters. The 3-foot orthoconic nautiloids of the shallows were successful precisely because they were not 30-foot behemoths like Camaraceras. They could respond faster to changes, required less food to maintain, and could exploit temporary food sources that larger predators could not survive on. Fossil evidence shows these shallow water nautiloids were extremely abundant. Some limestone formations consist largely of their shells, thousands of individuals preserved together, suggesting mass mortality events where entire populations died simultaneously. These events might have been caused by sudden temperature changes, toxic algae blooms, or storms that damaged too many shells for the nautiloids to survive. The abundance also indicates these were successful animals, populating shallow seas in huge numbers for millions of years. By the Devonian period, competition from armored fish and early sharks began reducing orthoconic nautiloid diversity. The straight shell design that served them well in earlier seas could not compete with the speed and maneuverability of fish with proper fins and streamlined bodies. Coiled nautiloids and ammonites began replacing the straight forms, their compact shells offering better hydrodynamics and more efficient buoyancy control. But for over 100 million years, orthoconic nautiloids proved that a simple geometric shape could be a devastating hunting tool if used correctly. The spear-like shell remains one of evolution's more elegant solutions to the problem of being a slow predator in fast waters. Point downward, adjust depth, wait for prey to swim into striking range, attack with tentacles before they realize the danger. Simple, effective, deadly. The shallow water spears hunted in bright sunlight, visible to everything around them, and still dominated their ecosystems. Sometimes the best camouflage is not hiding. Sometimes the best camouflage is being so perfectly positioned that prey do not recognize the threat until tentacles wrap around them and drag them into a waiting beak. Anomalocarids dominated the Cambrian seas, then vanished as prey evolved better defenses and new predators took over their ecological roles. Except one lineage refused to go extinct. Schinderhannes lived during the Devonian period, 150 million years after Anomalocaris should have disappeared. While the ocean had moved on to sharks, placoderms, and sophisticated jawed fish, this creature still hunted using the same frontal appendages and circular mouth its Cambrian ancestors pioneered. Scientists could not believe the fossil when they first examined it. The frontal grasping appendages were unmistakable, spine structures identical to Anomalocaris in design and function. The body showed the same flexible lobes for swimming. Even the head structure matched the Cambrian predator's basic layout. But the rock formation dates to the early Devonian, an era when Anomalocarids were supposed to be extinct. Either the dating was wrong, or evolution had played a stranger game than anyone expected. Additional specimens confirmed the impossible. Schinderhannes was a genuine Anomalocarid living in Devonian seas, surrounded by predators that should have outcompeted it into extinction ages ago. The creature measured about 4 inches long, much smaller than its Cambrian relatives, possibly an adaptation that allowed it to survive by exploiting niches the larger Devonian predators ignored. The discovery forced scientists to reconsider what they thought they knew about extinction and survival. Anomalocarids were supposed to be evolutionary dead ends, body plans that worked briefly during the Cambrian explosion but failed when faced with more advanced predators. Schinderhannes proved that assessment wrong. The design could survive and compete even in ecosystems dominated by jawed fish and heavily armored placoderms. How did it manage this? The small size was crucial. At 4 inches, Schinderhannes could hunt prey that larger predators could not efficiently catch. Tiny arthropods, worms, and other invertebrates provided food sources that Dunkleosteus and its relatives ignored. By specializing in small prey, Schinderhannes avoided direct competition with the apex predators sharing its environment. The frontal appendages still worked perfectly for grasping prey. While jawed fish could bite and hold, they struggled with very small, fast-moving targets. The appendages Schinderhannes inherited from its Cambrian ancestors were precision instruments, capable of plucking individual prey items from the water with accuracy jaws could not match. The spines held struggling prey firmly while the circular mouth processed the food. Schinderhannes also lived in a specific environment that may have offered protection. The fossils come from the Hunsrück slate in Germany, deposits that formed in relatively deep, low-oxygen waters. These conditions might have limited the number of large predators that could survive there, creating a refuge where Schinderhannes could persist without facing the full competitive pressure of shallow water ecosystems. The discovery raises an important question about extinction. How many other supposedly extinct lineages might have survived in isolated environments or ecological niches we have not examined closely? The fossil record is incomplete, biased toward animals that lived in conditions favorable for fossilization. Creatures living in environments with poor preservation potential could survive for millions of years without leaving evidence we can find. Schinderhannes might not have been alone. There could have been other Anomalocarid survivors in other parts of the world, in habitats that did not preserve fossils well. We might never find evidence of them, but that does not mean they did not exist. The gaps in our knowledge of Paleozoic life are enormous, and every new discovery reveals how much we still do not understand. What we know is Schinderhannes proved the Anomalocarid body plan was more versatile than scientists believed. It could adapt to new environments, new prey, new competitors. It could shrink down to exploit niches others ignored. It could survive when everything suggested it should have gone extinct with its Cambrian relatives. Eventually, Schinderhannes did go extinct, probably during one of the Devonian extinction events that killed off many marine species. But it lasted far longer than it had any right to. A living fossil swimming through seas that had forgotten what its ancestors looked like. The lesson is simple but important. Extinction is not always immediate. Lineages can persist in marginal environments, reduced populations, isolated refugees. What looks like a dead end might just be a detour lasting millions of years. Schinderhannes was supposed to be impossible. It existed anyway. Sometimes survival is just stubbornness with evolutionary time to prove the point. It stands 2 feet tall, rooted to the seafloor, branches spreading in perfect geometric patterns. Each branch subdivides into smaller branches, which subdivide again, creating a fractal structure that repeats at every scale. The symmetry is flawless, the organization mathematical, the design unlike anything produced by conventional biology. This is Charniodiscus, and scientists still debate whether it was even an animal. The organism lived during the Ediacaran period, the time just before the Cambrian explosion, when life was experimenting with multicellular body plans. Most Ediacaran organisms looked alien, lacking the bilateral symmetry and clear organ systems that would later define animal life. Charniodiscus was among the strangest of these strange creatures. The fractal branching pattern resembles nothing in modern biology. Animals grow through cell division and differentiation, processes that rarely produce perfect mathematical symmetry. Plants can show fractal-like branching, but plants use photosynthesis and have internal structures Charniodiscus lacked. Fungi sometimes display fractal growth patterns, but fungi decompose organic matter and show different tissue organization than what fossilized Charniodiscus displays. What was it? Some scientists classify Charniodiscus as an early animal, possibly related to sea pens, modern animals that look superficially similar and also grow in branching colonies. Sea pens are cnidarians, relatives of jellyfish and corals, and they feed by filtering plankton from water currents using stinging cells on their branches. If Charniodiscus was related to sea pens, it would represent an early attempt at colonial animal life, multiple individual polyps working together as a single organism. But other scientists argue Charniodiscus was not an animal at all. The fractal structure might indicate a completely different form of life, one that grew through processes we no longer see in modern organisms. Some proposals suggest Charniodiscus could have been a quilted organism, a body plan based on inflated chambers arranged in patterns that distributed stress evenly across the structure. This quilted construction would have allowed Charniodiscus to grow large without developing complex organ systems. The quilted hypothesis explains several strange features. Charniodiscus fossils show no evidence of a mouth, gut, or circulatory system. There are no feeding structures visible, no excretory organs, nothing that suggests the organism processed food the way animals do. Instead, the entire outer covering might have absorbed nutrients directly from the water, similar to how bacteria feed, but at a much larger scale. If true, this would make Charniodiscus a failed evolutionary experiment, an organism that tried a completely different approach to multicellular life, one that worked briefly during the Ediacaran period, but could not compete once true animals with guts and mouths evolved. The quilted body plan would have been too limited, unable to adapt to the complex predator-prey dynamics that emerged during the Cambrian explosion. Some researchers go further, suggesting Charniodiscus might not have been a single organism. Instead, it could have been a colonial structure built by microbes, similar to stromatolites, but with a more complex architecture. The fractal branching would have maximized surface area for the microbial colonies, allowing them to capture nutrients more efficiently from the water flowing past. This hypothesis is controversial because it reduces Charniodiscus from a sophisticated organism to essentially a bacterial apartment complex. The fossil evidence does not clearly support any single interpretation. Charniodiscus left impressions in soft seafloor mud, preserving the external structure but providing no information about internal anatomy or feeding mechanisms. We can see what it looked like, but not how it lived, what it ate, or even if it was truly alive in the sense we normally use that word. What we know is Charniodiscus was successful for a time. Fossils appear in formations across the world, from Newfoundland to Australia to Russia. The organism grew in shallow seas where water currents provided steady flow, suggesting it did require nutrients from the surrounding environment. The populations must have been substantial because the fossils are common in the formations where they appear. Then, the Cambrian explosion arrived with its mobile predators and active grazers. Charniodiscus vanished entirely, along with most other Ediacaran organisms. The fractal body plan that worked in the quiet Ediacaran seas could not survive in the dynamic Cambrian environment where animals moved, hunted, and competed actively. Whatever Charniodiscus was, it represents a fundamentally different approach to multicellular life. An approach evolution tried and abandoned, leaving behind only impressions in stone and endless questions about what we are looking at when we study those fossils. Was it an animal? A plant? A fungus? A colony? Something completely different from any modern form of life? We might never know with certainty. Some mysteries stay mysterious despite our best efforts to solve them. Charniodiscus stands in the fossil record as a reminder that life has tried forms we can barely imagine, and most of those experiments failed. The ones that succeeded became us. The fish swims with its head tilted downward, the wide boomerang-shaped skull cutting through water like a hydrofoil. Cephalaspis was a jawless fish that lived during the Silurian and Devonian periods, distinguished by a head shield so bizarrely shaped that scientists have spent over a century arguing about what it was for. The shield extends far to the sides, creating wings that make the head wider than the entire body is long. Why would evolution produce such an extreme design? The early theory suggested the head shield was armor, protection from predators like sea scorpions and early-jawed fish. Bone is heavy and requires energy to grow and maintain, but if predation pressure was high enough, the investment might be worth the protection. The shield covered the top and sides of the head completely, leaving only the underside exposed. But that explanation has problems. If the shield was purely defensive, why make it so wide? A compact armored head would provide better protection with less weight and drag. The extreme width of Cephalaspis' head shield actually made it more visible to predators and harder to maneuver in tight spaces. Pure defense does not explain the boomerang shape. The second theory focused on hydrodynamics. The wide head shield might have generated lift as water flowed over it, similar to how airplane wings create lift by directing airflow. This would allow Cephalaspis to glide above the seafloor while feeding, reducing the energy needed to maintain position. The fish could tilt its head down, creating an angle of attack that pushed water under the shield and generated upward force. This theory explains the shape better than simple armor protection. Cephalaspis lived on or near the seafloor, feeding on organic material it sucked up through its jawless mouth. The ability to hover just above the bottom would let it cover more ground while feeding, increasing efficiency. The wide head shield acting as a hydrofoil makes functional sense for this lifestyle, but scientists still debate the details. Some argue the shield was too small to generate significant lift, especially given the weight of the bone. Others point out that even small amounts of lift would reduce the energy cost of swimming, making the adaptation worthwhile. Still others suggest the shield might have served multiple purposes, combining some armor protection with hydrodynamic benefits. Recent studies using computational fluid dynamics tested how water would flow over a Cephalaspis head shield. The results were surprising. At certain angles and speeds, the shield did generate measurable lift, but it also created significant drag that would slow the fish down. The net benefit depended on exactly how Cephalaspis swam and fed, variables we cannot determine from fossils alone. The sensory organs on the head shield add another layer of complexity. Cephalaspis had areas on the shield covered with small polygonal scales that appear to be sensory fields, possibly for detecting water pressure changes or electrical signals from prey. These fields were positioned on the underside of the shield's wings, exactly where they would be if the fish was detecting prey hiding in seafloor sediment while hovering above. This suggests Cephalaspis was an active hunter, not just a passive filter feeder. The sensory fields would let it detect buried prey. The hovering ability would let it patrol efficiently, and the jawless mouth could suck up prey once located. The entire head shield design starts to make sense as an integrated feeding system rather than a single-purpose structure. But we still cannot be completely certain. Cephalaspis left abundant fossils showing the head shield in detail, but behavior does not fossilize. We can hypothesize how it used that bizarre head shape, test our ideas with fluid dynamics models, compare it to modern bottom-feeding fish. The evidence supports the hydrofoil plus sensors interpretation better than pure armor protection. Yet doubt remains because evolution is creative in ways we struggle to predict. The head shield might have served purposes we have not even considered. Display for attracting mates, acoustic resonance for communication, thermoregulation in variable water temperatures, species recognition to distinguish Cephalaspis from similar-looking jawless fish. Without time machines or living specimens, we are guessing based on physics and comparison to modern animals. What we know for certain is the boomerang head worked well enough that Cephalaspis thrived for millions of years. Multiple species evolved, each with slight variations on the head shield design. The fish spread across Devonian seas, leaving fossils in formations from North America to Europe to Asia. This was not a rare oddity barely surviving in marginal environments. This was a successful design that dominated seafloor feeding niches. Then, jawed fish evolved better ways to feed and fight, and the armored jawless fish went extinct. But the boomerang heads remain in stone, still confusing paleontologists, still resisting simple explanations. Sometimes, the strangest designs are the hardest to understand, even when we think we have figured them out. The dorsal fin is flat on top like a table, covered in tooth-like scales called denticles that point forward. Below the fin, on the shark's head, sits another patch of forward-pointing denticles forming a brush-like structure. When Stethocanthus tilted its head up, the two patches would face each other, creating a bizarre double brush arrangement that scientists have struggled to explain for decades. This was a shark that looked like it was designed by the committee, each part added without consulting the others. Stethocanthus lived during the Devonian and Carboniferous periods, reaching lengths of about 3 feet. The body was typical shark anatomy, sleek and muscular with proper fins and a powerful tail. Nothing unusual except for that head and dorsal fin arrangement that makes no obvious sense from a predation or defense perspective. The flat-topped dorsal fin especially confuses researchers. Dorsal fins normally serve as stabilizers, preventing the shark from rolling while swimming. A triangular fin works perfectly for this purpose. Flattening the top reduces the fin's effectiveness as a stabilizer while adding no obvious benefit. The denticles pointing forward create drag instead of reducing it. So why evolve such a strange modification? The most popular theory suggests the structure was for display, specifically for male-to-male competition or courtship. Many modern fish have elaborate fins used purely for attracting mates or intimidating rivals. The peacock bass with its humped back, the sailfish with its massive dorsal fin, the betta fish with flowing fins that serve no purpose except looking impressive. Sexual selection can drive the evolution of structures that seem counterproductive from a survival standpoint. If Stethocanthus used its dorsal fin and head brush for display, the arrangement starts to make sense. The shark could approach rivals or potential mates head-on, tilting its head up to present both brushed areas simultaneously. The forward-pointing denticles would catch light differently than smooth skin, creating visual contrast that made the display more visible. The unusual silhouette would be instantly recognizable to other Stethocanthus, serving as species identification. But this is speculation based on modern fish behavior, not direct evidence. We have no way to know if Stethocanthus actually displayed, if only males had the elaborate fins, if the structure was used in combat or courtship, or something else entirely. The fossils show the anatomy, but not the behavior that would explain it. Some scientists suggest the brush structures were sensory organs. The denticles might have been sensitive to water movement, helping Stethocanthus detect prey or predators. Arranging them in dense patches on the head and dorsal fin would create sensitive zones that could pick up vibrations from different directions. This would give the shark enhanced awareness of its surroundings. The problem with this theory is that sharks already have excellent sensory systems. They can detect electrical fields with specialized organs. They can smell blood in extremely diluted concentrations. They have lateral lines running along their bodies detecting water pressure changes. Adding more sensory patches seems redundant when they already have multiple sophisticated detection systems. A third hypothesis suggests the structures were defensive. If a predator tried to bite Stethocanthus from above, it would encounter the denticle brush on the dorsal fin first. The forward-pointing denticles could damage the predator's mouth or get stuck in teeth, making Stethocanthus difficult to swallow. This passive defense would not require any action from the shark beyond having the structures present. This explanation has merit, but does not address why the head also has a denticle patch. Predators attacking from above would not encounter the head brush unless they were trying to swallow the shark head first, an unlikely scenario given the shark's ability to swim and maneuver. The truth might combine elements of all three theories. The dorsal fin structure could provide some defense while also serving as a visual display, and the denticles might have minor sensory functions as a bonus. Evolution rarely produces single-purpose structures. Most anatomical features serve multiple roles, and the exact balance of functions can be difficult to determine from fossils alone. What we know is Stethecanthus was successful, despite carrying these bizarre structures. The shark lived for tens of millions of years, long enough to spread across multiple ocean basins and leave fossils in formations on different continents. The ironing board fin and head brush did not prevent survival and reproduction, which means the structures were either beneficial or at least not harmful enough to be selected against. Stethecanthus went extinct during the Carboniferous period, replaced by more conventional sharks. The elaborate dorsal fin disappeared from the shark lineage completely, never to evolve again. Modern sharks have varied dorsal fins, but none have flattened tops covered in forward-pointing denticles. Whatever advantage the structure provided was not worth keeping in later shark evolution. The fossils remain as evidence of an experiment evolution tried and abandoned. A shark with an ironing board on its back and a brush on its head, swimming through ancient seas for reasons we can only guess. Sometimes the strangest features have the simplest explanations, and sometimes they just stay strange. The pectoral fins have become jointed appendages covered in armored plates, resembling crab claws more than fish fins. Bothrialepis could walk on these modified fins, pulling itself along the seafloor or through shallow waters where swimming was less efficient than crawling. This was an armored placoderm that abandoned conventional fish movement for a hybrid approach combining swimming, walking, and possibly even brief excursions onto land. The claws were mechanical marvels. Each joint was precisely articulated, allowing controlled movement in multiple directions. The armor plates overlapped like roof shingles, protecting the internal muscles while permitting a flexibility. The claws could grip substrates, push against rocks, and manipulate objects with surprising dexterity for a fish. Scientists believe Bothrialepis used these appendages primarily for feeding. The fish spent most of its time on the bottom of rivers and lakes, using the clawed fins to dig through sediment looking for food. The heavily armored head acted as a plow, pushing into mud while the claws pulled the fish forward and excavated material. The small, weak jaws sucked up organic debris and small invertebrates exposed by the digging. This feeding strategy explains the extreme armor. Bothrialepis lived in environments with limited visibility, murky waters where sediment stayed suspended constantly. The armor protected the fish from collisions with rocks and debris while digging. The claws prevented the fins from being damaged by abrasive substrates. The entire body plan was optimized for bottom feeding in harsh conditions, but the armor created problems. Bothrialepis was front-heavy, with most of the body weight concentrated in the armored head and pectoral region. The tail was flexible and relatively light, providing some swimming propulsion, but not enough to make Bothrialepis fast or agile. In open water, the fish would have been slow and vulnerable to predators. This is why Bothrialepis stayed near the bottom and in shallow waters. The environment provided places to hide among rocks and vegetation. The sediment-rich water reduced visibility for predators. The abundance of organic material in muddy substrates provided constant food sources. Bothrialepis had found a niche where its specialized body plan was an advantage rather than a liability. The clawed fins also enabled something unusual for fish. Bothrialepis could potentially move between pools during droughts or low water periods. If the fins were strong enough to support the fish's weight briefly, it could walk short distances over exposed mud or rocks to reach new water sources. This ability would have been crucial in freshwater environments where seasonal changes could isolate pools temporarily. Fossil evidence supports this possibility. Bothriolapis fossils are common in ancient river and lake deposits, environments subject to seasonal drying. The widespread distribution across multiple continents suggests Bothriolapis could disperse effectively, possibly by walking overland when necessary. Some trackways preserved in stone might have been made by Bothriolapis, though definitive proof is difficult without finding a fossil directly associated with the tracks. The fish was extremely successful by any measure. Bothriolapis fossils are among the most abundant of all placoderms, found in huge numbers in Devonian formations worldwide. Some deposits contain hundreds of individuals preserved together, suggesting mass mortality events where entire populations died simultaneously, possibly from sudden drought or toxic algae blooms. The abundance indicates Bothriolapis filled its ecological niche effectively. The combination of armor, specialized feeding appendages, and semi-terrestrial capability allowed it to exploit resources other fish could not access. In rivers and lakes across Devonian continents, Bothriolapis dominated as the primary bottom feeder, but the specialized design was also a limitation. When environmental conditions changed at the end of the Devonian period, Bothriolapis could not adapt quickly enough. The heavy armor and specialized feeding strategy that worked perfectly in shallow, sediment-rich environments became disadvantages in clearer, deeper waters. More streamlined fish with better swimming ability outcompeted Bothriolapis in the changing ecosystems. The entire placoderm group went extinct, including every Bothriolapis species. The crab claw fins disappeared completely from the fish lineage, never to evolve again in that exact form. Modern fish have developed various modified pectoral fins for bottom walking, like mudskippers and frogfish, but none match the jointed armored appendages Bothriolapis pioneered. The fossils still show those claws in perfect detail, joints visible, armor plates clearly defined. Looking at them, you can almost see how they moved, how they gripped and pulled, how they helped a strange armored fish crawl through ancient mud in search of food. Sometimes evolution creates solutions so specific they only work in exact circumstances. When those circumstances disappear, the solutions become useless, and the creatures carrying them go extinct. The claw closes around a fish half its own length, serrated edges slicing through scales and flesh. Behind the claw comes 7 feet of segmented arthropod body, paddle-like swimming appendages rowing through brackish water where rivers meet the sea. Compound eyes on stalks track movement in the murky transition zone. This is Pterygotus, the largest arthropod that ever lived, an apex predator that ruled estuaries for millions of years. Sea scorpions came in many sizes, but Pterygotus was the giant. The claws alone measured over a foot long, equipped with spines designed to grip struggling prey firmly. The serrated inner edges could saw through fish and other arthropods with ease. Once those claws clamped down, escape was nearly impossible. The prey would be dragged to waiting mandibles that could shred tissue efficiently. Estuaries were perfect hunting grounds for Pterygotus. The mixture of fresh and salt water created ecosystems packed with life. Fish migrated through estuaries between ocean feeding grounds and river spawning areas. Smaller arthropods thrived in the nutrient-rich waters. Vegetation grew densely along the edges, providing cover for ambush predators. Pterygotus exploited all these features. The animal could tolerate both fresh and salt water, allowing it to hunt in environments other predators avoided. The murky water reduced visibility, making ambush easier. The dense vegetation provided hiding spots where Pterygotus could wait for prey to swim past. The migration routes meant steady supplies of fish moving through the hunting grounds seasonally. The size of Pterygotus was both advantage and constraint. 7 feet of arthropod requires enormous amounts of food to fuel. The exoskeleton had to be thick enough to support that length without collapsing under the animal's own weight. Molting became increasingly dangerous as Pterygotus grew, each shed leaving it vulnerable and soft for extended periods. But the size also meant Pterygotus had almost no natural predators. Few animals in Devonian estuaries could threaten a 7-foot armored arthropod with foot-long claws. The largest placoderm fish might have been capable of killing Pterygotus, but they typically hunted in deeper waters rather than shallow estuaries. In its preferred habitat, Pterygotus was effectively invincible. The swimming method combined rowing and undulation. The paddle-like appendages provided thrust for forward movement, while the flexible segmented body created additional propulsion through wave-like motions. This hybrid approach was not as efficient as the streamlined swimming of fish, but it worked. Well enough for an ambush predator that relied on short bursts of speed rather than sustained pursuit. Fossil evidence shows Pterygotus could also walk on land, at least briefly. Trackways preserved in ancient mud show arthropod footprints matching Pterygotus's leg structure, leading from one body of water to another. The animal could apparently leave the water and cross short distances overland, possibly to reach isolated pools during dry seasons or to escape deteriorating water conditions. This semi-terrestrial capability gave Pterygotus options other purely aquatic predators lacked. If prey became scarce in one estuary, Pterygotus could potentially travel overland to find new hunting grounds. If environmental conditions became unfavorable, it could move to different water bodies. The combination of size, armor, and terrestrial ability made Pterygotus extraordinarily adaptable. The eyes deserve special mention. Compound eyes on movable stalks provided vision in multiple directions, simultaneously. Each eye contained thousands of individual lenses, creating detailed mosaic images of the surroundings. The eyes could rotate independently, letting Pterygotus watch for prey while monitoring for threats from other directions. In murky estuary waters where visibility was limited, superior vision was crucial. Pterygotus needed to spot prey before the prey spotted it. The eye stalks allowed precise positioning of the eyes above or around cover, letting the predator scout while remaining mostly hidden. This gave it significant advantages over prey species with fixed eyes, providing more limited fields of view. Despite its success, Pterygotus shared the fate of all sea scorpions. The group went extinct during the Permian period, unable to survive the environmental catastrophe that killed most ocean life. The size that made Pterygotus dominant also made it vulnerable. Large animals need more food, reproduce more slowly, and have smaller population sizes than smaller animals. When the food web collapsed, the giants died first. But for tens of millions of years, Pterygotus proved that arthropods could compete with vertebrates for apex predator status. 7 feet of armored nightmare with serrated claws and compound eyes, hunting where rivers met the sea. Occasional walks onto land showing the first hints of the terrestrial invasion arthropods would eventually complete with scorpions and spiders. The fossils still impress. Shells so large museums display them next to vertebrate predators for size comparison. And the arthropods hold their own in that comparison. For sheer intimidation factor, Pterygotus matched anything swimming in Devonian seas. Proof that evolution does not care whether you have a spine, as long as you can hunt effectively. The worm walks on legs covered in jointed armor. 10 pairs of appendages that look like they belong on an arthropod, not a soft-bodied creature. Dionea challenges everything scientists thought they understood about the evolution of arthropods and their distinctive jointed legs. This was a worm with armored walking appendages appearing in Cambrian seas before arthropods had fully established their body plan, suggesting the evolution of legs was more complex and experimental than previously believed. The fossil shows the details clearly. Each leg has multiple segments covered in hardened plates, creating a jointed structure that could bend and move in controlled ways. The body itself remains soft and worm-like, lacking the rigid exoskeleton that defines arthropods. This combination of soft body and hard legs seems contradictory, like evolution could not decide which direction to take. Scientists initially thought the fossil was wrongly assembled or misidentified. How could a worm have arthropod-like legs? The two features do not go together in modern animals. Worms have soft, unjointed appendages or no appendages at all. Arthropods have jointed legs and rigid exoskeletons covering their entire bodies. Dionea appeared to be a hybrid that should not exist, but additional fossils confirmed the bizarre anatomy. Dionea was real, and it was walking through Cambrian seas on armored legs, while the rest of its body remained soft and flexible. The arrangement suggests legs evolved before full body armor, contradicting the theory that arthropods developed hard exoskeletons first and then modified them into jointed appendages. This has massive implications for understanding arthropod evolution. If legs appeared before complete exoskeletons, then the evolutionary path from soft-bodied worms to hard-shelled arthropods was more gradual and experimental than scientists assumed. There might have been many intermediate forms, testing different combinations of soft and hard body parts, trying various arrangements before settling on the full arthropod design. Dionea was one of those experiments. The armored legs provided mobility and stability for walking on the seafloor. The soft body retained flexibility that rigid armor would have eliminated. The combination allowed Dionea to navigate complex terrain while maintaining the ability to squeeze through narrow spaces or flex around obstacles. But the design had limitations. The soft body was vulnerable to predation. Without armor protecting vital organs, any predator with sharp appendages or jaws could easily damage Dionea. The armored legs were heavy and required energy to move, creating metabolic costs the soft body might struggle to support. These limitations probably explain why Dionea and similar creatures did not persist. Evolution eventually favored full arthropod armor over partial leg armor. The protection and structural support provided by complete exoskeletons outweighed the flexibility advantages of soft bodies. By the mid-Cambrian period, true arthropods with fully armored bodies dominated, and the Dionea-style hybrids vanished. But Dionea's existence proves evolution tests many paths simultaneously. During the Cambrian explosion, body plans were still fluid and experimental. The rules defining modern animal groups had not solidified yet. Creatures could have features that seem contradictory by modern standards because those standards did not exist. Evolution was throwing everything at the wall to see what stuck. Most experiments failed, including Dionea's armored legs on soft body approach. The creatures that succeeded were the ones that found body plans flexible enough to adapt to changing conditions, but stable enough to pass on to descendants. Full arthropod armor with jointed legs provided that balance. Dionea's hybrid approach did not. The fossil record shows several other lobopodians, the group Dionea belongs to, with various combinations of soft bodies and modified appendages. Hallucigenia had rigid spines for defense. Others had different arrangements of legs, spines, and soft tissue. All were experimenting with ways to survive in Cambrian seas. Almost all failed except the lineages that led to modern arthropods and velvet worms. Dionea walked on armored legs through ancient oceans, testing an evolutionary path that ultimately led nowhere. The genes controlling leg armor might have been passed down to descendants that used them differently, eventually contributing to full arthropod exoskeletons. Or Dionea might have been a complete dead end, a lineage that left no descendants. We cannot know for certain because the fossil record has too many gaps. What we can say is Dionea showed legs could evolve independently of body armor, that evolution does not always proceed in the linear paths we imagine, that the Cambrian period was stranger and more experimental than even optimistic scientists assumed. The worm with armored legs walked briefly through history, proving that evolution tries every possible combination. Most combinations fail, but the failures are often more interesting than the successes. They show us what did not work and force us to reconsider what we think we know about how life evolves. Sometimes the dead ends teach us more than the survivors. The shell covers only the belly, leaving the back completely exposed. This is Anomalocaris, a turtle ancestor that evolution tried and rejected, a creature that shows how the iconic turtle shell developed through stages that modern turtles have abandoned completely. Modern turtles have shells covering both top and bottom, creating a protective box that shields them from most predators. The top shell, called the carapace, and bottom shell, called the plastron, connect along the sides, leaving only openings for the head, legs, and tail. Anomalocaris had only the plastron, the bottom shell, fully developed. The back was covered with thickened ribs that were beginning to fuse and expand, showing the first stages of what would become the carapace in later turtles. But the process was incomplete, leaving the back vulnerable to attack from above. Any predator biting down from above would encounter unprotected tissue and bone rather than hardened shell. This seems like a fatal flaw in design. Why would evolution produce a half shell that protects from below, but not from above? The answer lies in understanding where Anomalocaris lived and how it moved through its environment. The creature was semi-aquatic, spending time both in water and on land. In water, predators often attack from below, coming up from deeper areas to strike at prey from underneath. The belly shell protected Anomalocaris from this attack angle effectively. On land, the turtle could retreat to water when threatened from above. The partial shell was also lighter than a full shell would be. Carrying less weight made Anomalocaris faster and more agile than fully shelled turtles. This mattered in environments where escape speed could mean survival. A lighter animal could run faster on land, swim faster in water, and required less food to fuel its metabolism. But the disadvantages eventually outweighed the benefits. As predators evolved more sophisticated hunting strategies and stronger jaws, the partial shell provided insufficient protection. Turtles with complete shells survived predation better and passed on genes for full armor to their descendants. The half-shell approach that Anomalocaris pioneered was abandoned as evolution favored more complete protection. The fossil shows other interesting features beyond the incomplete shell. Anomalocaris had teeth, something modern turtles lack completely. The teeth were small and simple, probably used for gripping prey rather than chewing. Modern turtles evolved beaks made of keratin, the same material as fingernails and bird beaks, and lost their teeth entirely. The transition from toothed Anomalocaris to beaked modern turtles required millions of years of evolutionary refinement. The ribs on the back show clear evidence of the fusion process that would eventually create the carapace. In normal vertebrates, ribs are separate bones that curve around the body cavity, protecting organs while allowing flexibility. In turtles, the ribs fused together and expanded into broad plates, creating the solid shell structure. Anomalocaris shows this process halfway complete, with ribs that are thicker and wider than normal, but not yet fully fused into a solid covering. This intermediate stage reveals how evolution works through gradual modifications. The turtle shell did not appear suddenly in one dramatic evolutionary leap. It developed through countless small changes over millions of years, each change providing some benefit that helped the animal survive and reproduce. Anomalocaris represents one snapshot in that long process, showing what partial shell development looked like. Scientists found Anomalocaris fossils in late Triassic deposits, roughly 220 million years ago. This was after the Permian extinction, but before turtles had perfected their modern form. The environment was recovering from the extinction, with many ecological niches still open for colonization. Anomalocaris and other semi-shelled turtles were competing to fill those niches. Evolution chose the fully shelled versions. By the Jurassic period, turtles with complete carapaces and plastrons dominated, and the half-shelled forms went extinct. The extra weight and reduced agility of full shells were worth the improved protection. Modern turtles inherited that full shell design and have maintained it for over 200 million years. Anomalocaris walked and swam with a belly shell and no back armor, testing whether partial protection was enough. The answer was no. But the experiment taught us how shells develop, revealing the step-by-step process that created one of evolution's most distinctive defensive structures. Sometimes, the failures are more instructive than the successes. They show us what evolution tried and rejected, illuminating the path from simple ancestors to complex modern forms. Anomalocaris failed to survive, but it succeeded in teaching us how turtles became turtles. That is worth remembering, even if the half-shell design is gone forever. The teeth are not designed for biting once and swallowing. These teeth grip, tear, and hold struggling prey while the fish thrashes like a crocodile performing a death roll. Rhizodonts were lobe-finned fish that grew up to 23 feet long and hunted in freshwater rivers and lakes during the Carboniferous period. While most fish teeth are relatively simple cones designed for basic puncturing or grasping, rhizodont teeth were elaborate structures with multiple cusps, sharp edges, and recurved points that prevented prey from escaping. The tooth design reveals sophisticated predation strategies. Each tooth had a main cusp flanked by smaller accessory cusps, creating a serrated edge that could saw through flesh and bone. The teeth were also recurved, curving backward toward the throat, so any prey trying to pull away would only impale itself more deeply. Once a rhizodont bit down, the prey was effectively trapped. But teeth alone do not explain rhizodont success. The lobe fins were the real innovation. Unlike the ray fins of most fish, which are flexible but relatively weak, lobe fins have a muscular base supported by bones. This gives them more strength and precision for controlled movement. Rhizodonts use these powerful fins to hold position in fast-moving currents, to maneuver through complex underwater terrain, and possibly to perform the death rolls that made their feeding strategy so effective. The death roll hypothesis comes from tooth wear patterns. Rhizodont teeth show unusual wear that suggests the fish twisted violently while holding prey. This twisting motion would use the serrated teeth like saws, cutting through tissue more effectively than simple biting. Modern crocodiles use the exact same strategy, gripping prey and spinning to tear off chunks they can swallow. Rhizodonts might have been doing this 300 million years before crocodiles existed. The fossil evidence supports the parallel evolution of similar hunting techniques in completely unrelated animals. When a feeding strategy works well, evolution tends to rediscover it in different lineages. The combination of strong jaws, specialized teeth, and body rotation for tearing prey is simply too effective to ignore. Rhizodonts lived in freshwater ecosystems, dominated by amphibians and early reptiles. The competition for food was fierce, with multiple predators hunting the same prey species. Rhizodonts competed by growing large and developing teeth that could handle prey other predators struggled with. The ability to grip and tear allowed rhizodonts to eat animals with tough skin or armor that simple biting could not penetrate. Some rhizodont species specialized in hunting other fish, including smaller rhizodonts. Cannibalism was apparently common, with larger individuals eating smaller members of their own species. This is typical of apex predators in environments where large prey is abundant, but requires significant effort to catch and kill. Eating your own species is energy efficient because you are consuming animals with similar nutritional value to yourself. The lobe fins also hint at an evolutionary possibility that never materialized. Lobe-finned fish are the ancestors of all land vertebrates. The bones in lobe fins are homologous to the bones in your arms and legs, modified and adapted for terrestrial life. Rhizodonts had powerful lobe fins that could have potentially supported walking on land if evolution had pushed them in that direction. It did not. Rhizodonts remained fully aquatic predators. Other lobe-finned fish like Eusthenopteron and Tiktaalik made the transition to land, evolving into the first tetrapods. Rhizodonts stayed in the water, perfecting their aquatic hunting strategies instead of experimenting with terrestrial living. By the end of the Carboniferous period, rhizodonts were declining. Environmental changes reduced the freshwater habitats they dominated. Competition from early reptiles and sharks increased. The specialized teeth and body plan that made rhizodonts successful in Carboniferous rivers became disadvantages in changing ecosystems. The last rhizodonts went extinct in the early Permian, leaving no descendants. The lobe fins and specialized teeth disappeared from freshwater predator niches, replaced by reptiles that could hunt both in water and on land. The death roll strategy survived, but in crocodilian lineages rather than fish. Rhizodonts showed that fish could develop sophisticated predation techniques, rivaling anything land animals later evolved. Gripping teeth, powerful fins, twisting body motions to tear prey apart. These strategies work whether you are a fish or a reptile or a mammal. Evolution keeps rediscovering effective solutions because effectiveness transcends taxonomy. The teeth remain in the fossil record, still sharp, still showing the wear patterns from violent feeding, still teaching us that predation is an ancient art, refined over hundreds of millions of years by countless lineages testing what works. And sometimes, what works in fish works just as well in crocodiles. The eyes are larger than the rest of the head combined, bulging from the front of the carapace like biological searchlights. Thylacocephalans were arthropods that lived from the Cambrian through the Cretaceous periods, distinguished by eyes so disproportionately large that scientists still cannot explain what they needed that much visual capability for. These were not tiny creatures squinting at faint light in the deep ocean. These were active swimmers in relatively shallow waters where light was abundant. So why eyes that large? The body plan adds to the mystery. Thylacocephalans had a shield-like carapace covering the head and thorax, multiple pairs of raptorial appendages folded beneath the body like those of mantis, shrimp, and a flexible abdomen with swimming structures. The combination suggests active predation, but the huge eyes seem excessive for hunting prey in well-lit environments. Modern animals with proportionally large eyes usually live in extreme environments. Deep-sea fish have enormous eyes to gather minimal light in complete darkness. Nocturnal animals have large eyes for hunting at night. Thylacocephalans lived in neither environment, yet they evolved eyes that would make sense only for creatures needing maximum light-gathering capability. One theory suggests thylacocephalans hunted bioluminescent prey. If their prey used bioluminescence for communication or camouflage, large eyes with high sensitivity to light would help detect those signals. The thylacocephalan could spot glowing prey from greater distances and track them more effectively than predators with normal-sized eyes. This theory has problems though. Bioluminescence is most common in deep water where sunlight does not penetrate. Thylacocephalans lived in shallow to mid-depth waters where bioluminescence provides less advantage. The fossils are found in deposits that represent well-lit environments, not the dark depths where bioluminescence is crucial for survival. A second theory focuses on visual acuity rather than light sensitivity. Maybe thylacocephalans needed extremely sharp vision to hunt fast-moving prey in complex environments. Large eyes with more photoreceptors can create higher resolution images, helping predators track prey that dodges and weaves between obstacles. Modern dragonflies have enormous compound eyes, providing the visual acuity needed to catch flying insects in midair. Thylacocephalans might have been the dragonflies of ancient seas, hunting small, fast-moving prey that required superior vision to catch. This theory fits better with the fossil evidence, but it still does not fully explain eyes quite that large. Moderate enlargement would provide most of the visual acuity benefits without the metabolic costs of maintaining such massive organs. The raptorial appendages suggest ambush predation similar to mantis shrimp. Modern mantis shrimp have excellent vision, including the ability to see polarized light, and potentially more color channels than any other animal. They use this vision to identify prey, communicate with each other, and navigate complex reef environments. Perhaps thylakocephalans had similar visual capabilities we cannot detect in fossils. Soft tissue rarely preserves eye internal structures. We can see the size and general shape of thylakocephalan eyes, but we cannot determine if they had special adaptations for polarized light detection, UV sensitivity, or rapid motion tracking. All we know is the eyes were enormous relative to body size, implying vision was extremely important to their survival strategy. The fossil record shows thylakocephalans persisted for over 400 million years. This longevity proves the body plan, including the oversized eyes, was successful despite the metabolic costs involved. Eyes require constant energy to maintain, and larger eyes require proportionally more energy. Thylakocephalans must have obtained enough food to justify the investment. The group finally went extinct at the end of the Cretaceous period, the same extinction event that killed the dinosaurs. Whatever advantages the huge eyes provided were not sufficient to survive the environmental catastrophe that ended the Mesozoic era. The thylakocephalans vanished along with countless other marine species, ending a lineage that had seen the rise and fall of trilobites, the evolution of fish, and the dominance of marine reptiles. The fossils still show those enormous eyes, still suggesting mysteries we cannot solve completely. Why did these creatures need such powerful vision? What were they seeing that required biological searchlights mounted on their heads? What advantage justified the energy cost of maintaining eyes that large for hundreds of millions of years? We may never know with certainty. Some questions about extinct animals cannot be answered conclusively because the critical information does not fossilize. Behavior, soft tissue structures, environmental context, all can be lost to time, leaving only tantalizing hints of capabilities we can measure, but not fully understand. Thylakocephalans swam through ancient seas with eyes bigger than their heads, seeing things we can only imagine. The eyes remain their most striking feature and their most persistent mystery. The fish is pressed flat as paper, the entire body barely thicker than your thumb, but spanning nearly a foot wide. Drepanaspis was a jawless fish that lived during the Devonian period, distinguished by a body shape so extremely flattened that scientists initially thought the fossils were compressed by geological pressure during fossilization. They were not compressed. Drepanaspis really was that flat. The extreme body plan was an adaptation to life in murky, sediment-heavy waters. Drepanaspis lived in shallow seas and estuaries where visibility was minimal and conventional hunting by sight was nearly impossible. The fish spent most of its time on the bottom, vacuum feeding by sucking sediment into its mouth and filtering out organic particles. The flat body allowed it to glide over soft mud without sinking, essentially hovering just above the seafloor while feeding. The mouth was positioned on the underside of the head, perfect for bottom feeding. As Drepanaspis moved forward, the mouth would intake sediment continuously, passing it through gill rakers that filtered out food particles while expelling clean water through the gills. This feeding strategy required no vision, no complex prey capture behaviors, just constant forward movement and efficient filtration. The head was heavily armored with bony plates. These plates protected Drepanaspis from predators attacking from above, the most likely attack angle for a bottom-dwelling fish. The armor also helped the fish push through dense vegetation and debris on the seafloor, without damaging soft tissue. The combination of flat body and armored head created a living plow that could excavate food from challenging environments, but the flat body had significant disadvantages. Swimming efficiency was poor because the pancake shape created massive drag in the water. Drepanaspis could not swim fast or maneuver quickly, making it vulnerable to pursuit predators. The fish compensated by staying in murky water where predators could not see well, and by relying on armor to survive any attacks that did occur. The eyes were small and positioned on top of the head. This placement suggests Drepanaspis did not rely heavily on vision for feeding, but did need to watch for predators approaching from above. The small eye size indicates limited visual acuity, probably only enough to detect movement and changes in light levels rather than forming detailed images. Drepanaspis belonged to the group called heterostracans, jawless fish that experimented with various body shapes during the Paleozoic era. Some heterostracans were streamlined and fast, others were heavily armored and slow. Drepanaspis represented the extreme flatfish strategy, reducing body thickness to the minimum possible while maintaining vital organ function. The strategy worked well in specific environments. Drepanaspis fossils are abundant in formations representing shallow, murky waters with soft sediment bottoms. These were environments where competition from jawed fish was lower because the murky water and soft substrates favoured specialised bottom feeders over active hunters. But as water clarity improved and jawed fish evolved better sensory systems, the environmental niches Drepanaspis dominated became accessible to competitors. Placoderms like Bothriolopis could dig through sediment more effectively with their specialised appendages. Early sharks could hunt in murky water using electroreception and lateral line systems. The advantages that made Drepanaspis successful in early Devonian seas became less relevant as ecosystems evolved. The fossil record shows Drepanaspis declining through the Devonian period before going extinct entirely. The extremely flat body plan was abandoned by evolution, never appearing again in quite the same form. Modern flatfish like flounders and rays have flattened bodies, but they achieved flatness through different anatomical modifications and used different feeding strategies. Drepanaspis proved that extreme specialization can be successful temporarily, but creates vulnerabilities that eventually lead to extinction. The pancake fish dominated muddy bottom environments for millions of years by being perfectly adapted to conditions other fish struggled with. When those conditions changed, or when competitors evolved better adaptations, Drepanaspis had nowhere to go. The flat body that made it successful prevented it from adapting to new circumstances. This is a pattern that appears repeatedly in the fossil record. Highly specialized organisms dominate specific niches, but lack the flexibility to survive when environments change. Generalist species that can adapt to varied conditions tend to persist longer, even if they never dominate as completely as specialists in optimal environments. Drepanaspis was a specialist taken to an extreme, flat as paper, armor-plated, vacuum feeding in murky waters where nothing else could compete effectively. Perfect for its niche. Unable to exist outside that niche. When the niche disappeared, so did Drepanaspis. The lesson applies beyond extinct fish. Specialization is powerful, but risky. Adaptability matters more than perfection when the only constant is change. The ocean was dying. Not slowly, not gradually, but catastrophically. Losing species faster than evolution could replace them. 96% of all marine species went extinct in roughly 1 million years, the worst mass extinction in Earth's history. The Permian period ended not with a whimper, but with environmental collapse on a scale that makes modern climate concerns look trivial. What caused it? The evidence points to massive volcanic eruptions in Siberia, events so prolonged and violent that they covered an area larger than Europe, with lava flows up to 2 miles thick. These eruptions lasted for hundreds of thousands of years, pumping carbon dioxide, methane, and toxic gases into the atmosphere at rates that overwhelmed natural carbon cycles. The atmospheric changes triggered cascading disasters. Global temperatures rose by 10 to 15 degrees Fahrenheit. Ocean temperatures increased even more dramatically because water retains heat longer than air. The warming oceans held less dissolved oxygen, creating anoxic zones where most animals could not survive. Acidification from absorbed carbon dioxide dissolved calcium carbonate, destroying the shells of countless marine organisms. But the killing mechanisms went beyond simple warming and oxygen depletion. The Siberian eruptions vaporized massive coal deposits, releasing mercury and other heavy metals that poisoned ecosystems. Methane hydrates on the seafloor destabilized as temperatures rose, releasing even more greenhouse gases in a runaway feedback loop. Hydrogen sulfide produced by bacteria in anoxic waters accumulated to toxic levels, possibly even reaching the atmosphere. The combination was lethal. Trilobites, survivors of 4 previous mass extinctions, went completely extinct. Every single species vanished, ending a lineage that had dominated seafloors for 270 million years. Brachiopods that built reef cities for 300 million years died by the thousands of species. Rugose and tabulate corals went extinct entirely. Even single-celled organisms like Foraminifera experienced catastrophic losses. The reef ecosystems collapsed completely. Coral reefs that had existed for hundreds of millions of years died and did not return for millions of years afterward. The ecological roles they filled remained empty, with no organisms capable of building similar structures. The ocean became a biological desert in many areas, with severely reduced biodiversity and simplified food webs. On land, the extinction was nearly as severe. 70% of terrestrial vertebrate species died. Forests burned or died from temperature stress. Lakes and rivers turned toxic from acidification. The few survivors faced a world fundamentally different from the one they evolved in, with resources scarce and competition minimal among the remaining species. Recovery took millions of years. The early Triassic period shows severely reduced diversity across all ecosystems. The few survivors reproduced and diversified slowly, hindered by continued environmental instability and low oxygen levels that persisted long after the main extinction pulse. Some ecological niches remained, unfilled for 5 to 10 million years before new organisms evolved to occupy them. The extinction reset evolution completely. Groups that dominated Permian seas like brachiopods and rugose corals never recovered their former diversity. New groups like modern corals and mollusks took over the vacated niches, establishing the marine ecosystems we recognize today. On land, synapsids that had dominated were replaced by archosaurs that would eventually give rise to dinosaurs. The Permian extinction demonstrates how fragile complex ecosystems are. Hundreds of millions of years of evolution, countless successful species, elaborate food webs and ecological relationships, all erased in geologically brief time by environmental changes the organisms could not adapt to quickly enough. The speed of the extinction was the critical factor. If the same changes had occurred over 10 million years instead of 1 million, many more species might have survived by evolving tolerance to new conditions. But 1 million years is not enough time for evolution to work. Most species have generation times measured in months or years, not days. Developing tolerance to 10-degree temperature changes, severe oxygen depletion, and ocean acidification requires genetic changes that only accumulate over thousands of generations. The Permian extinction moved faster than evolution could respond. The survivors were mostly generalists. Species adapted to variable conditions survive better than specialists. Animals capable of tolerating wide temperature ranges, low oxygen levels, and varied food sources had advantages over organisms with narrow environmental requirements. This pattern appears in most mass extinctions. Generalists survive, specialists die. The modern relevance is unavoidable. Current climate change involves temperature increases and ocean acidification occurring at rates comparable to the Permian extinction, though not yet at the same magnitude. Carbon dioxide levels are rising, faster than at any time in the geological record except during mass extinction events. Ocean pH is dropping as carbonate chemistry shifts. The difference is we have the data to see it coming. The Permian extinction happened to organisms with no ability to understand or predict environmental changes. They could only react to conditions that were already lethal. We have the advantage of science, showing us where current trends lead if uncorrected. Whether that advantage matters depends on whether we use it. The Permian Ocean died because volcanic eruptions nobody could control pumped carbon into the atmosphere faster than natural systems could handle. Our carbon emissions are doing the same thing, just from different sources. The ocean did recover eventually, Life did diversify again. New ecosystems did develop. But 96% of species did not survive to see that recovery. They died in oceans that turned toxic, that lost oxygen, that became too hot to support the life that had thrived there for hundreds of millions of years. The fossils remain as evidence of what happens when environments change faster than evolution can adapt. The lesson is simple. Survival requires adapting to change before the change becomes catastrophic. Once the catastrophe starts, it is already too late for most species. 500 million years of evolutionary experimentation compressed into fossil layers we can hold in our hands. Every creature in this story died. Every species went extinct. The trilobites with their crystal eyes, the placoderms with their guillotine jaws, the sea scorpions that walked on land, all vanished completely. The ocean that created them no longer exists. The ecosystems they dominated are gone, but the lessons remain. These animals teach us that dominance is temporary. Trilobites ruled for 270 million years, longer than dinosaurs would exist, longer than mammals have existed so far. Their reign seemed permanent, but it was not. When conditions changed, the animals that had succeeded for hundreds of millions of years died as quickly as any other species. Success does not guarantee survival. Anomalocaris was the perfect predator for Cambrian seas, with vision and grasping appendages unmatched by anything else alive. It went extinct when prey evolved defenses and new predators appeared. Dunkleosteus had the most powerful bite in Devonian oceans, bone guillotines that could shear through armor. It went extinct when ecosystems favored speed over strength. Adaptation matters more than perfection. Schinderhannes survived 150 million years after its kind should have died by adapting to new niches. Drepanaspis thrived by specializing in murky waters where competitors struggled. Both strategies worked temporarily, but both ultimately failed when environments changed beyond their adaptive capacity. Specialization is powerful but fragile. The flat body that made Drepanaspis successful in muddy shallows prevented it from adapting to clearer waters. The heavy armor that protected Bothriolepis made it too slow to escape faster predators. Every adaptation that increases success in one environment creates vulnerability in different conditions. The Permian extinction proved that no amount of evolutionary success protects against catastrophic environmental change. Trilobites survived 4 previous mass extinctions. They had proven their ability to persist through environmental disasters that killed countless other species. But the 5th extinction was too severe, too fast, too comprehensive. Even the ultimate survivors could not survive when 96% of the ocean died. These lessons apply to modern life, including us. Humans are the dominant species on Earth now, shaping ecosystems globally, extracting resources at rates no other species can match. We have technology, intelligence, and adaptability that seem to guarantee our survival, but the Paleozoic creatures had their own guarantees. Trilobites had crystal eyes and armor. Dunkleosteus had unstoppable jaws. Anomalocaris had size and power. All were dominant. All went extinct. The ocean does not care about dominance. It only cares about what can survive current conditions. When conditions change fast enough, previous success becomes irrelevant. The creatures best adapted to the old environment die first. The survivors are the ones that can tolerate new conditions, regardless of how well they performed before. We are changing conditions rapidly now, pumping carbon dioxide into the atmosphere at rates that match geological catastrophes. Ocean acidification is accelerating. Temperature records break yearly. The changes happening in decades normally require thousands or millions of years. The Paleozoic creatures did not cause their own extinctions. We are causing ours. Volcanic eruptions killed the Permian Ocean. We cannot stop volcanoes, but we can stop our carbon emissions, can reduce acidification, can slow the changes we are creating. The difference between us and Anomalocaris is we understand what is happening and can change course. Whether we will change course is different from whether we can. The fossil record suggests most species do not change course. They keep doing what worked until it stops working, then they go extinct. Adaptation requires changing behavior before the consequences force change, and most organisms cannot do that. But we are not most organisms. We have science, we have historical records, we have fossil evidence showing exactly what happens when environments change faster than evolution adapts. We can learn from 500 million years of other species' mistakes. The question is whether we will. Every creature in this story was perfectly adapted to its environment until the environment changed. Then perfect adaptations became fatal specializations. The animals could not change fast enough and died. We can change fast enough. We have the technology, the knowledge, the capability to adapt our civilization to sustainable patterns before catastrophic climate change makes that adaptation impossible. The dead teach us that waiting too long means joining them. Trilobites did not wait. They could not. They had no choice but to face environmental changes as they occurred. We have choices. We can see what is coming and act before it arrives. The ocean that killed 96% of its species did eventually recover. Life did return. New ecosystems did develop, more sophisticated and diverse than what existed before. But that recovery took millions of years, and it did not include any of the species that dominated the Permian Ocean. If we fail, the ocean will recover without us. Life will continue, diversify, evolve into new forms. The planet will be fine eventually, but we will not be here to see it. The Paleozoic creatures never knew they were leaving lessons. They just lived, hunted, bred, and died. Their fossils teach us by accident, preserved coincidences that reveal what survival requires and what causes extinction. The lessons are clear. Dominance is temporary. Specialization is fragile. Adaptation matters more than perfection. Environmental change kills faster than evolution can respond. Success does not guarantee survival. We are living in their ocean now, changed and modified, but ultimately the same system that created trilobites and killed them. The rules have not changed. Species that cannot adapt go extinct. Ecosystems that lose too many species collapse. Recovery takes millions of years. The dead have spoken across 500 million years. The living should listen. Because becoming fossils ourselves would make this the shortest-lived dominant species in Earth's history. And we have better options than that. We just need to choose them before it is too late. The ocean will not wait. It never does.